You all to Karnataka CET and NEET coaching program organized by Department of Pre-University Education, Karnataka. My name is Devaraju R. In this session, I will be discussing synapses and CET questions pertaining to second PU chapters Human Reproduction and Reproductive Health. Human beings are unisexual and exhibit sexual dimorphism. Sexual dimorphism is the existence of both male and female organisms in nature. The characters which distinguishes them are called secondary sexual characters. The male and female reproductive systems are distinct in order to bring about the coordination between male and female with respect to sequential events in sexual reproduction such as gametogenesis, gamete transfer or insemination, formation of zygote, implantation of embryo, organogenesis, parturition, lactation, etc. Reproductive system consists of two types of sex organs, namely the primary sex organs and the secondary sex organs. Primary sex organs are those they produce gametes and sex hormones. Sex organs that are concerned with the production of gametes and sex hormones are called primary sex organs. Examples would be gonads. The male gonad is called testis and the female gonad is called ovary. Testis are the male gonads which are concerned with the production of a male gamete called sperms or spermatozoans and secretion of a male sex hormone called testosterone. So testosterone is very much essential for the expression of secondary sexual characters in males. Similarly, so ovary are concerned with the production of a female gamete called agar ovum and secretions of two sex hormones, namely estrogen and progesterone. Estrogen is responsible for the expression of secondary sexual characters in females, whereas progesterone is concerned with the maintenance of pregnancy, so therefore it is called a pregnancy hormone. Secondary sex organs are not concerned with the production of gametes and sex hormones, but they are essential in sexual reproduction as they help in transport of gametes to the site of fertilization. Examples for secondary sex organs include system of ducts, accessory glands, and external genitalia associated with the male and female reproductive system. Let us take up the various aspects of male and female reproductive system one by one. Male reproductive system. Male reproductive system consists of a pair of testes, which are the primary sex organs. System of ducts includes retitestis, vasa efferentia, epididymis, then vasa deferens, also called sperm duct, then ejaculatory duct, urethra or urinogenital duct. So these are the ducts associated with male reproductive system. And accessory glands of the male reproductive system includes a pair of seminal vesicles, a single prostate gland, and a pair of bulbourethral or corpus glands. External genitalia includes scrotum and penis. A pair of ovaries which are oval shaped, each measuring about 4 cm, are located outside the abdomen in a bag or sac-like structure called scrotum. That's why they are said to be extra abdominal in position. So TS of testis reveals that each testis is made up of numerous testicular lobules. There are about 250 testicular lobules in each testis, and the long and highly connected tubules present in these lobules are called seminiferous tubules. So seminiferous tubules are the structural and functional unit of testis. The interstitial cells located between these tubules are called Leydig cells, which secretes a male androgen or sex hormone called testosterone. The epithelial cells lining these seminiferous tubules is called germinal epithelium. So germinal epithelium is made up of two groups of cells, namely spermatogenic cells or spermatogonia, so which on spermatogenesis gives rise to sperms. And second group of cells are sustentacular cells or supportive cells, also called Sertoli cells. So named after an Italian physiologist, Sertoli. So they provide necessary nutrition for the development of germ cells. So that's the reason why Sertoli cells are also called nerve cells. You can see in the diagram itself. So this is a spermatid. The heads of the spermatids are embedded in the Sertoli cells to get their nutrition. So this is the reason why we call Sertoli cells as a nutritive cells or nerve cells. System of ducts. As I told you, testis is made up of numerous testicular lobules. So these testicular lobules consist of structural and functional units. We call them as seminiferous tubules. So all the seminiferous tubules of a testis opens into a network. So this network is called retitestis. So retitestis joined and rejoined to form 10 to 12 tubules like structures. We call them as vasa efferentia. So vasa efferentia open into epididymis. So epididymis is a long uncoiled tube of about 6 meters in length, where sperms are stored temporarily and ready for ejaculation. So maturation of sperms also takes place in the epididymis. So for this reason, epididymis is considered as the storehouse of the sperms itself. So then epididymis opens into vas deferens. Vas deferens of both the testes enter into abdomen, where they loops over the urinary bladder and they receive ducts from the seminal vesicles and form a duct. The duct is known as ejaculatory duct. Ejaculatory duct passes through the middle of the urethra and opens at the tip of the penis, called urethral meatus. What is the significance of this duct system? As I told you, the system of ducts is a part of male reproductive system. It is said to be secondary sex organ, so helps in the transport of sperms or gametes into the site of fertilization, so during coitus or sexual intercourse. So let us take a quick recap. Path of movement of sperms in the duct systems during ejaculation. So nimgella gottide, so testis alli there are numerous lobules. ಆ lobules ನಾವು ಏನಂತ ಕರಿತೀವಿ ಅಂದ್ರೆ ಟೆಸ್ಟಿಕ್ಯುಲರ್ lobules ಅಂತ ಕರಿತೀವಿ. ಈ ಟೆಸ್ಟಿಕ್ಯುಲರ್ lobules ಅಲ್ಲಿ ಇರತಕ್ಕಂತ ಫಂಕ್ಷನಲ್ ಯೂನಿಟ್ಸ್ ಗೆ ನಾವು seminiferous tubules ಅಂತ ಕರಿತೀವಿ. So seminiferous tubules are the structural and functional unit of testis, where spermatogenesis takes place, and the sperm cell production ಆಗೋದು ಈ seminiferous tubules ಅಲ್ಲಿ. So seminiferous tubules of the testis open into network. ಆ ಒಂದು ನೆಟ್ವರ್ಕ್ ನಾವೇಂತ ಕರಿತೀವಿ, retitestis ಅಂತ ಕರಿತೀವಿ. Then sperms and seminiferous tubules enter into retitestis. So from there they enter into vasa efferentia. So vasa efferentia ಇಂದ epididymis ಹೋಗುತ್ತೆ. Epididymis ಇಂದ vas deferens ಹೋಗುತ್ತೆ, and from vas deferens sperms enter into ejaculatory duct, and you know that ejaculatory duct passes through the middle of the urethra and finally opens at the tip of the penis during ejaculation at the time of sexual intercourse. So this is the diagram, and this is a representation showing the path of movements of sperms in the duct system, especially during ejaculation. Coming to accessory glands. Accessory glands of the male reproductive system includes a pair of seminal vesicles, a prostate gland, and a pair of bulbourethral glands, also called corpus glands. The secretions of these glands is called seminal plasma. Seminal plasma is mixed with sperms in the duct system. As a result, it provides fluid medium or living medium for the sperms itself. That's very essential. Then it provides energy for the movement of sperms. It acts as a lubricant during sexual intercourse, and it provides alkaline medium for the removal of acidity present in the male and female genital tracts during sexual intercourse. ನಾನು ಹೇಳ್ತಾ ಇದ್ದೆ ಈಗ ಸೆಮಿನಲ್ ಪ್ಲಾಸ್ಮಾ ಅನ್ನೋದು ಈ ಅಕ್ಸೆಸರಿ ಗ್ಲ್ಯಾಂಡ್ಸ್ ಇಂದ ಸೆಕ್ರೀಟ್ ತಗೋದೊಂದು ಫ್ಲೂಯಿಡ್. The secretions of accessory glands is called seminal plasma. It's mixed with duct systems, ಅಂದ್ರೆ it mixed with sperms in the duct system and it forms semen. ಇಲ್ಲಿ semen ಅಂತ ಹೇಳಿದ್ರೆ ಎರಡನ್ನೂ ಸೇರಿಸಿ ನಾವು semen ಅಂತ ಕರಿತೀವಿ. So sperms along with the seminal plasma is called as semen. Together constitute seminal plasma, sperms together constitute semen. What is the composition of the seminal plasma or semen? Semen is rich in fructose for nourishing sperms. It is rich in fibrinogen, a clotting factor for forming coagulum inside vagina. We know that when sperms are released into the vagina, the sperm cells clot like blood cells. This is known as coagulum. Formation of coagulation requires this fibrinogen. So after some time, coagulum dissolves with the help of an enzyme called fibrinolysins. So fibrinolysins helps in dissolving coagulum present inside the vagina. The very important aspects of seminal plasma is the presence of prostaglandins. So prostaglandins are required for thinning of cervical mucus and induction of uterine contractions for upward movement of sperms in the female genital tract. It also composed of calcium and bicarbonate ions for providing alkaline pH to neutralize acidity of male and female genital tracts during sexual intercourse. So these are the components or constituents of semen, which includes sperms and seminal plasma. Coming to male external genitalia. So male external genitalia includes scrotum and penis. So these are the parts of male reproductive system considered external genitalia. Penis is the male copulatory organ which is used to inseminate the female during sexual intercourse. It's a cylindrical and erectile organ. It is made up of a central shaft consisting 3 longitudinal columns of spongy tissue, 2 dorsolateral columns called corpora cavernosa, and a ventral column known as corpus spongiosum. In the center of the corpus spongiosum itself, this urethra opens out. Nodi, idu structure of a penis, idu scrotum, okay, where testicles are present. So idune ನಾವು ಏನು ಕರಿತೀವಿ ಅಂತ ಹೇಳಿದ್ರೆ, so columns of it's a columns of ಸ್ಪಾಂಜಿ ಟಿಶ್ಯೂ ಅಂತ ಕರಿತೀವಿ. ಆ 3 ಕಾಲಮ್ಸ್ ಇರುತ್ತೆ. ಆ 3 ಕಾಲಮ್ಸ್ ಅಲ್ಲಿ ಡಾರ್ಸೋಲ್ಯಾಟ್ರಲ್ ಅಂತ ಏನು ಕರಿತೀವಿ, ಮೇಲ್ಗಡೆ ಇರ್ತಕ್ಕಂಥ ಕಾಲಮ್ಸ್ಗೆ ನಾವು ಕಾರ್ಪೊರಾ ಕೆವರ್ನೋಸ್ ಅಂತ ಕರಿತೀವಿ. ಸೊ ಇಲ್ಲಿ ವೆಂಟ್ರಲ್ ಕಾಲಮ್ಗೆ ನಾವು ಕಾರ್ಪಸ್ ಸ್ಪಾಂಜಿಯೋಸಮ್ ಅಂತ ಕರಿತೀವಿ. ಈ ಒಂದು ರೀಜನ್ಸ್ ಅಲ್ಲಿ ಈ ಯುರೇಥ್ರಾ ಓಪನ್ಸ್ ಔಟ್. Coming to the very important aspects of male reproductive system, that is spermatogenesis. Spermatogenesis is the process of formation of sperms. You know that sperms are formed from primordial germ cells present in the testis, particularly seminiferous tubules itself, and where spermatogenesis takes place. Primordial germ cells, before they transformed into spermatozoa or sperms, pass through 3 different phases. The first one is called multiplication phase. It involves mitosis. The second phase is called growth phase, and the third phase is called maturation phase. It involves some meiosis, that is first meiosis and second meiosis. Primordial germ cells undergo repeated mitotic cell division, so results in the formation of a numerous cells. These cells are called spermatogonia, also called sperm mother cells. So sperm mother cells undergo growth phase. That means they grow in size. The growth of the spermatogonia is due to accumulation of cytoplasm and even due to replication of DNA itself. Such enlarged cells are called as primary spermatocytes. Now, primary spermatocytes undergo maturation. As I told you, it involves meiosis. So primary spermatocytes undergo first meiosis, results in the formation of secondary spermatocytes. Now, secondary spermatocytes undergo second meiosis, results in the formation of spermatids, and finally spermatids are transformed into sperms by a process called spermiogenesis. So spermiogenesis is a differentiation process which transforms non-functional spermatids into sperms. But you should remember, here primordial germ cells are diploid in nature, so spermatogonia or sperm mother cells are diploid in nature. Okay, and finally, so it gives rise to haploid sperms. So this is the product of spermatogenesis. So as a result of spermatogenesis, each primary spermatocyte gives rise to 4 sperms. Look at the structure of human sperm. Human sperms are minute, microscopic, haploid male gametes formed in the testis during spermatogenesis. Each sperm is covered over by plasma membrane and distinguished into 4 parts. The one is called head region, then the next region is neck region, then the middle piece and the tail. These are the 4 regions of the sperm. Head is the anterior part of the sperm or proximal part of the sperm. So it is somewhat oval shaped. Over the head there is a cap-like structure. This is known as acrosome. So this acrosome is filled with enzymes. These are known as hyaluronidase enzyme. So plays a vital role at the time of fertilization. So they're meant for dissolving egg membranes during fertilization. So therefore, acrosome helps the sperm to penetrate into the ovum during fertilization. Then the neck region. So the function of neck region is to connect the head with the middle piece. So neck region consists of 2 centrioles, proximal centriole and distal centriole. So distal centriole continues with the axial filament or it attaches to axial filament. Then the middle piece is the middle part of the sperm. So in the middle part, you can see axial filament is surrounded by spirally coiled mitochondria. So these are known as nebenkern. The cytoplasmic sheath around the nebenkern is highly reduced. So this is known as manchet. The mitochondria present in the middle piece provide necessary energy for the movement of sperms in the female genital tracts during fertilization. Then the next region is called tail. So tail is the posterior or distal part of the sperm. distinguished into main piece and end piece. So what is main piece then? The part of the tail where axial filament which is surrounded by cytoplasmic sheath is known as main piece, and the part of the tail where only axial filament is present without any cytoplasmic sheath is called end piece. The significance of tail is that it helps the sperm to move or swim in the female genital tract during fertilization. So coming to hormonal control of spermatogenesis. Spermatogenesis is under the control of gonadotrophin releasing hormones secreted by hypothalamus. So gonadotrophin releasing hormones act on anterior lobe of pituitary for the secretion of two gonadotrophins, namely LH and FSH. LH is luteinizing hormone and FSH, follicle stimulating hormone, is also a gonadotrophin. So LH and FSH both are said to be gonadotrophins because they act on gonads. So what is the significance of LH? LH acts on Leydig cells, interstitial cells of the testis known as Leydig cells. So induce Leydig cells to secrete testosterone, and this testosterone is responsible for the process called spermatogenesis to occur. Similarly, follicle stimulating hormone F or FSH acts on Sertoli cells of the testis, induce them to secrete certain factors which are responsible for the transformation of spermatids into sperm. So this is known as spermiogenesis or spermateliosis. So this is a regulation of, this is a representation showing the regulation of spermatogenesis by hormones. Coming to female reproductive system. Female reproductive system consists of a pair of ovaries which are the primary sex organs. Then it includes system of ducts. System of ducts includes fallopian tubes, uterus, cervix and vagina. Accessory glands are associated with the vestibule. They are vestibular glands. External genitalia of the female reproductive system includes vagina along with a pair of mammary glands also called breasts. Ovaries are present in the pelvic region. They are attached to the wall of the uterus and abdomen by means of ligaments. Each ovary is distinguished into outer cortex and inner medulla. Cortex is lined by germinal epithelium, whereas medulla consists of connective tissue stroma. So in the connective tissue stroma, which is rich in blood vessels, blood cells as well as nerve endings. In the stroma region itself, there are numerous ovarian follicles. So these ovarian follicles are present at different stages of their maturity. Some are at primary follicular stage, some are at secondary follicular stage, some are at tertiary follicular stage. There is also a mature follicle. The mature follicle present in the ovary is called Graafian follicle. So ovum is present in the center of the Graafian follicle. When the ovum matures, Graafian follicle ruptures, releasing ovum. So this process is called ovulation. So ovulation is the process of release of egg or ovum from the Graafian follicle. So it occurs in the middle of the menstrual cycle or on the 14th day of the menstrual cycle. After ovulation, the ruptured Graafian follicle is converted into corpus luteum. Corpus luteum starts secreting a sex hormone called progesterone in expectation of pregnancy and fertilization. If fertilization does not occur, corpus luteum undergo degeneration and it remains as a white scare in the ovary. So this white scare or white body is called or corpus albicans. So this is how the follicular cells mature in the ovary to produce a mature egg or ovum, and that undergo a process called ovulation to release ovum. So after ovulation, Graafian follicle is converted to corpus luteum in expectation of this pregnancy. Corpus luteum starts secreting progesterone. If fertilization does not occur, this undergo degeneration. So degenerated corpus luteum is said to be corpus albicans. So this is a white body. So this is a yellow body. There are differences with respect to spermatogenesis and oogenesis in case of human female. Spermatogenesis as well as spermatogenesis occurs in males and oogenesis occurs in female. So unlike males, where spermatogenesis begins on the onset of puberty, that is about 13 or 14 years onwards, but in females, oogenesis begins even before birth with the production of primary follicles. However, so these primary follicles mature into Graafian follicle on the onset of puberty. So these primary follicles mature into secondary follicles. Secondary follicles mature into tertiary follicle. So tertiary follicles mature into graphene follicle. So graphene follicle is nothing but the mature ovarian follicle present in the ovary. So you know that in the center of the graphene follicle, ovum or egg is present. When the egg matures, graphene follicle ruptures, releasing ovum. The process is called ovulation. So this released egg may undergo fertilization. So after ovulation, the ruptured graphene follicle is converted into another structure called corpus luteum. It's also called yellow body, and it starts secreting progesterone in anticipation of fertilization and pregnancy. If fertilization does not occur by 28 days or so, then corpus luteum undergo degeneration into a white body. This is known as corpus albicans. So ಈ ಒಂದು diagrammatic representation ಏನು ತೋರಿಸುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ the mature or maturation of follicles into egg or ovum. So these are the different stages of maturation of follicles in the ovary itself. Coming to graphene follicle. Graphene follicle is named after Dutch anatomist Reiner de Graaf. It's a mature follicle in the ovary. It is spherical in shape, covered over by an outer fibrous layer. This is known as theca externa. It is covered by inner layer called theca interna. Several layers of cells present below the theca interna constitute membrana granulosa. The cells of the membrana granulosa project towards one side, forming a mound of cells. This is known as cumulus ooporus. So this encloses secondary oocyte. The cavity present in the graphene follicle is called antrum. So antrum is the cavity present in the graphene follicle. So this is the stage at which ovulation occurs. When ovulation occurs, the ovum is at secondary oocyte stage. It has completed meiosis one only. So it completes its second meiosis only when sperm enters inside, so at the time of fertilization. Coming to system of ducts. System of ducts includes a pair of fallopian tubes, one for each ovary, then a single uterus. Both the fallopian tubes open into uterus. The cavity in the uterus is somewhat triangular shaped. It's also called womb of the mother. Then the wall of the uterus is differentiated into three layers, namely outer perimetrium, inner middle myometrium, and inner endometrium. The inner wall of the uterus is called endometrium. The inner lining of the uterus is the endometrium. So this is a site for implantation of embryo. The uterus opens into cervix. The mouth of the uterus is called cervix. So it opens into vagina through cervical canal. So cervix is the birth canal to which infant passes to be born. It also acts as a passage for menstrual flow during menstruation in females. External genital organs. External genitalia of the female reproductive system includes vagina. It is associated with a slit-like opening in the center. This is known as vestibule. So this is known as vestibule. So having consisting two openings, one is called urethral opening and the other one is called vaginal opening. Vestibule is surrounded by two thin fleshy folds of skin called labia minora. So labia minora is free from pubic hairs as well as sweat glands. Labia minora in turn covered by two thick fleshy folds of skin called labia majora. So these are covered by hairs, pubic hairs as well as sweat glands. You know that this is the clitoris, the place in the anterior region where two labia minora fuse. So there is a highly erectile tissue. This erectile tissue is called as clitoris, and it is the sensitive part of the female reproductive system. So below the abdomen there is a rounded protuberance. Due to accumulation of fat, this rounded protuberance is called as mons pubis. So this is the anus, and in case of virgin females, the opening of the vagina is guarded by perforated membranes. This is known as hymen. So hymen may get ruptured due to various activities of the female, maybe due to cycling, horseback riding, so taking part in sports activities, and also due to exercise. So therefore, the presence or absence of hymen is the not reliable indicator of virginity in females. So all these structures associated with vagina or external genitalia are collectively called as vulva. So vulva includes vestibule, a slit-like opening in the center, with the two openings called urethral opening and vaginal opening, and two folds of skin around the vestibule, labia minora, and two folds of skin surrounding the labia minora or majora, and clitoris and even mons pubis together constitute vulva. Mammary glands. Presence of mammary glands is the characteristic feature of mammals. So human females are able to feed their young ones or newborns with the help of milk produced in these mammary glands. In case of mammals, mammary glands are present as rounded structures. These rounded structures are called as breasts. Each breast is made up of a nipple. It is porous and it is used by the newborn for suckling. A circular pigmented area present below the nipple is called areola. It is made up of circular muscles. Each breast consists of considerable amount of fat and glandular tissues. The glandular tissues of the breast is divided into 15 to 20 lobes. So these lobes are called as mammary lobes. A cluster of milk secreting cells present in each lobe is called mammary alveoli. So they are meant for synthesis and storage of milk. The milk which is stored in between the spaces present between the alveolar cells or alveoli cells. The milk from alveoli flows into mammary tubules. From mammary tubules, it goes to mammary duct. So this is the mammary tubule. It goes to mammary duct. From mammary duct, it goes to mammary ampulla. From mammary ampulla, milk goes to lactiferous duct. So lactiferous duct opens outside at the tip of the nipple. So this is a structure of a breast showing the different aspects of it. So therefore, the flow or ejection of milk, so during breastfeeding the child by mother, starts from mammary lobes. Mammary lobes alli milk secreting cells idave, avana alveoli cells anta karitivi. So alveoli cells alli ಏನು ಮಾಡ್ತಾರೆ ಅಂತ ಹೇಳಿದ್ರೆ they produce milk. ಈ ಮಿಲ್ಕ್ ಆಲ್ವಿಯೋಲೈ ಸೆಲ್ಸ್ ಇಂದ ಎಲ್ಲಿಗೆ ಹೋಗುತ್ತೆ ಅಂದರೆ ಮ್ಯಾಮರಿ ಟ್ಯೂಬ್ಯೂಲ್ಸ್ಗೆ ಹೋಗುತ್ತೆ. ಮ್ಯಾಮರಿ ಟ್ಯೂಬ್ಯೂಲ್ಸ್ ಇಂದ ಎಲ್ಲಿಗೆ ಹೋಗ್ಬೇಕಂದ್ರೆ ಮ್ಯಾಮರಿ ಡಕ್ಸ್ಗೆ ಹೋಗುತ್ತೆ. ಯು ನೋ ದಟ್ ಇದೊಂದು ಟ್ಯೂಬ್ಯೂಲು, ಇದೊಂದು ಟ್ಯೂಬ್ಯೂಲ್. ಈ ಟ್ಯೂಬ್ಯೂಲ್ಸ್ ಎಲ್ಲ ಜಾಯ್ನ್ ಆಗಿ ಬರ್ತಕ್ಕಂಥ ಒಂದು ಸ್ಟ್ರಕ್ಚರ್ಗೆ ನಾವು ಮ್ಯಾಮರಿ ಡಕ್ಟ್ ಅಂತ ಕರಿತೀವಿ. ಮ್ಯಾಮರಿ ಡಕ್ಟ್ ಓಪನ್ಸ್ ಇಂಟು ಮ್ಯಾಮರಿ ಆಂಪ್ಯುಲಾ. ಮ್ಯಾಮರಿ ಆಂಪ್ಯುಲಾ ಓಪನ್ಸ್ ಇಂಟು ಲ್ಯಾಕ್ಟಿಫೆರಸ್ ಡಕ್ಟ್. ಈ ಲ್ಯಾಕ್ಟಿಫೆರಸ್ ಡಕ್ಟ್ ಅಥವಾ ಲ್ಯಾಕ್ಟಿಫೆರಸ್ ಡಕ್ಟ್ ಓಪನ್ಸ್ ಔಟ್ಸೈಡ್ ಅಟ್ ದ ಟಿಪ್ ಆಫ್ ದ nipple. As a result, so milk flows into the mouth of the child. So it is to be remembered that the production and ejection of milk is under the control of two hormones of the pituitary. The one is called prolactin and the other one is called oxytocin. Prolactin is responsible for synthesis of milk in the mammary glands, whereas oxytocin is responsible for ejection of milk. Coming to the female gamete called agar ovum. Agar ovum is spherical in shape. It is said to be aelicithal. It is called aelicithal because it is free from yolk. You know that yolk is a reserve food material that provides nutrition for the developing embryo. As we see in case of bird's eggs itself, since human egg, the development is internal. The development of the fetus or the development is internal. So developing fetus gets its nutrition directly from the mother through placenta. Therefore, there is no need of yolk. That's why human egg is free from yolk. This condition is called aelicithal. Unlike other eggs, for example, in case of birds, so bird's egg is surrounded by calcareous shell. But in case of human, calcareous shell is not present. That's why they are said to be non-cladoic. So this is the cytoplasm of the egg. It's also called ooplasm. There is a haploid nucleus. Cytoplasm is surrounded by primary plasma membrane, which in turn covered by acellular or non-cellular layer. So this is known as zona pellucida, which is similar to vitelline membranes of lower vertebrates. The space present between the plasma membrane and zona pellucida is called perivitelline space. The polar bodies released during meiosis 1 and 2 are present in the perivitelline space present between plasma membrane as well as zona pellucida. Zona pellucida in turn covered by epithelium with radiating projections. So these are known as corona radiata. This is the structure of a human egg. Coming to oogenesis. The process of formation of agar ovum in the ovary is called oogenesis. Egg is formed from primordial germ cells present in the ovary. So primordial germ cells, before they transform into agar ovum, pass through three different phases. namely multiplication phase, growth phase, and maturation phase. Primordial germ cells undergo repeated mitotic phase or repeated multiplication phase. It's also called proliferative phase. Results in the formation of large number of cells called oogonia, also called egg mother cell. Egg mother cell or oogonia undergo growth phase. So here is a in growth phase they grow in size. So due to accumulation of what you can say yolk, the process of production and storage of yolk is called vitellogenesis. It is to be remembered, synthesis of yolk called vitellogenesis is not observed in humans, as development is internal and the developing fetus gets its nutrition directly from the mother through placenta. So therefore, synthesis of yolk is insignificant in case of human egg. Nimge ಗೊತ್ತಿದೆ. So even in the developmental stage, mother is able to feed the fetus with help of placenta. Even after birth itself, mother is able to feed the newborn with help of milk produced in the mammary glands. So therefore, there is no need for vitellogenesis as far human egg is concerned. So that's why it is insignificant. Anyhow, the grown cells are called primary oocytes. They are diploid in nature. Primary oocytes undergo maturation phase. So primary oocytes undergo meiosis 1, it's unequal division. As a result, primary oocytes gives rise to one larger cell, it is called secondary oocyte, and a smaller body, this is known as first polar body or first polocyte. Both secondary oocyte and first polar body undergo second meiosis. Again, it's unequal division. As a result, secondary oocyte gives rise to one egg or ovum, it is larger inside, and one smaller body. Similarly, first polar body also undergo meiosis 2, results in the formation of two more polar bodies. As a result, each primary oocyte gives rise to one egg or ovum and totally three polar bodies. The egg or ovum undergo directly functions as a ovum, ooty directly functions as a ovum. There is no specialization process as we see in case of transformation of spermatids into sperm called spermiogenesis. Coming to menstrual cycle. From menses to menopause, the female reproductive system undergoes cyclic changes in anticipation of pregnancy and fertilization. So these changes are mainly concerned with uterus, that is endometrium, and also ovarian follicles or in the ovary itself. Every month, one egg is released from the alternative ovary between the successive months. When egg is released in the middle of the menstrual cycle, it may get fertilized. The fertilized egg is called zygote. Zygote undergo a process called embryogenesis. You know that what is embryogenesis? The process of formation of embryo from diploid zygote is called embryogenesis. So thereby moralized form, blastolized form, or blastocyst form. So meanwhile, uterus makes certain preparation and it waits for the blastocyst to come and attach to the wall of the endometrium. So if this is called implantation of embryo. If this does not occur by 28 day or so, the wall of the endometrium breaks and changes are repeated once again in the ovary to produce another egg. So this cycle of changes that occur in the female reproductive system during reproductive phase is called menstrual cycle. Menstrual cycle in human female is of 28 duration and can be studied under four main headings as menstruation phase, follicular or proliferative phase, then ovulatory phase, then luteal phase or secretory phase. The first phase of menstrual cycle is menstruation phase. It lasts for 1 to 5 days. The second phase is called follicular phase, lasts for about 6 to 12 days itself. And the third phase is ovulatory phase, occurs between 12 to 14 days. And then the last phase is called luteal phase or secretory phase, so between 16 to 28 days or so. So coming to menstrual phase, menstruation phase. So this is the first phase of menstruation during which the wall of the endometrium breaks. As a result, so menstrual flow occurs and it comes out of the uterus by the way of vagina. So this is during menstruation. The next phase is called follicular phase. It lasts for about 6 to 12 days. The first 2 days of the proliferative phase is used for the reconstruction of the endometrium that is lost during menstruation. It is called proliferative phase because the cells of the endometrium as well as the follicular cells undergo proliferation or multiplication, so-called proliferative phase. It is also called follicular phase because the ovarian follicles, primary follicles undergo maturation under the influence of FSH, follicle stimulating hormone secreted by anterior lobe of pituitary. So during follicular phase, say about 6 to 8 primary follicles undergo maturation under the influence of follicle stimulating hormone, of which only one develops into Graafian follicle. You know that what is Graafian follicle? It's a mature follicle present in the ovary. So the cells surrounding the Graafian follicle, you know that theca interna is surrounded by membrana granulosa. The cellular layer, the cell layer present below the theca interna is called membrana granulosa. The cells of the membrana granulosa secretes estrogen. That's why follicular phase is also called estrogen phase. During the same time, the levels of FSH and LH rises. Increased concentration or increased levels of LH is called LH surge. This LH surge is responsible for rupturing of this Graafian follicle, because you know that so in the center of the Graafian follicle, agar ovum is present. It is at secondary oocyte stage. So LH ಏನು ಮಾಡುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ, it is meant for rupturing of Graafian follicle. So this is the hormone which induces ovulation in human female. That means, so it is responsible for the rupturing of Graafian follicle. As a result, ovulation occurs. The process of release of agar ovum from the Graafian follicle is called ovulation. It occurs in the middle of the menstrual cycle, exactly on the 14th day of the menstrual cycle. So the next phase is called luteal phase or secretory phase. Here luteal phase refers to the process of formation of corpus luteum. After ovulation, the ruptured Graafian follicle is converted into corpus luteum. Now corpus luteum is maintained by LH. So LH maintains corpus luteum for continued secretion of a sex hormone called progesterone. You know that. So progesterone is responsible for maintenance of pregnancy. If fertilization does not occur, because ovulation ಆಗುತ್ತೆ, ovulation ಆದಮೇಲೆ ಅದು ಏನಾಗಬಹುದು, ಫೆಲೋಪಿಯನ್ ಟ್ಯೂಬ್ಗೆ ಎಂಟ್ರಿ ಆಗುತ್ತೆ. ಅದೇ ಟೈಮಲ್ಲಿ ಅದು ಸ್ಪರ್ಮ್ನ ಮೀಟ್ ಮಾಡಿದ್ರೆ ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗುತ್ತೆ. ಫರ್ಟಿಲೈಸೇಷನ್ ಆದರೆ ದಟ್ ಲೀಡ್ಸ್ ಟು ಪ್ರೆಗ್ನೆನ್ಸಿ. ಅವಾಗ ಏನು ಮಾಡುತ್ತೆ ಅಂದರೆ ಕಾರ್ಪಸ್ ಲ್ಯೂಟಿಯಂ ಸ್ಟಾರ್ಟ್ ಸೆಕ್ರೇಟಿಂಗ್ ಪ್ರೊಜೆಸ್ಟ್ರಾನ್. ಸೊ ಟಿಲ್ ದ ಕಂಪ್ಲೀಷನ್ ಆಫ್ ಪ್ರೆಗ್ನೆನ್ಸಿ, ಸೇ ಅಬೌಟ್ 9 months or so. ಆ ಕಾರ್ಪಸ್ ಲ್ಯೂಟಿಯಂ ಫಂಕ್ಷನ್ಸ್ ಆಗಬೇಕಾದ್ರೆ ನಮಗೆ LH ಬೇಕಾಗುತ್ತೆ, ಲ್ಯೂಟಿನೈಸಿಂಗ್ ಹಾರ್ಮೋನ್ ಬೇಕಾಗುತ್ತೆ. ಸೊ ದಿಸ್ ದ ಕಂಡೀಷನ್. ಇನ್ ಕೇಸ್ ಇಫ್ ಫರ್ಟಿಲೈಸೇಷನ್ ಡಸ್ ನಾಟ್ ಅಕ್ಕರ್, ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗದೆ ಇದ್ರೆ ಏನಾಗ್ಬೋದು ಅಂದರೆ ದ ಲೆವೆಲ್ಸ್ ಆಫ್ ಲ್ಯೂಟಿನೈಸಿಂಗ್ ಹಾರ್ಮೋನ್ ಆಟೋಮೆಟಿಕ್ಲಿ ಡಿಕ್ಲೈನ್ಸ್. ಯಾಕಂತ ಹೇಳಿದ್ರೆ ಕಾರ್ಪಸ್ ಲ್ಯೂಟಿಯಂನ ಮೇಂಟೈನ್ ಮಾಡೋ ಅವಶ್ಯಕತೆ ಬರೋದಿಲ್ಲ. ಅದಕ್ಕೋಸ್ಕರ LH ಲೆವೆಲ್ ಡಿಕ್ಲೈನ್ಸ್. LH ಲೆವೆಲ್ಸ್ ಡಿಕ್ಲೈನ್ ಆದಾಗ ಕಾರ್ಪಸ್ ಲ್ಯೂಟಿಯಂ ಕೆನಾಟ್ ಫಂಕ್ಷನ್. ಸೊ ಅವಾಗ ಏನಾಗುತ್ತೆ, ಕಾರ್ಪಸ್ ಲ್ಯೂಟಿಯಂ ಅಂಡರ್ಗೋ ಡಿಜನರೇಷನ್ ಇಂಟು ಕಾರ್ಪಸ್ ಆಲ್ಬಿಕನ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಇಟ್ಸ್ ಆಲ್ಸೋ ಕಾಲ್ಡ್ ವೈಟ್ ಬಾಡಿ. ಸೊ ಅವಾಗ ಏನಾಗುತ್ತೆ, ಪ್ರೊಜೆಸ್ಟ್ರಾನ್ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಆಟೋಮೆಟಿಕಲಿ ಡಿಕ್ಲೈನ್ಸ್. ದ ಇಮಿಡಿಯೇಟ್ decline in the concentration of progesterone is responsible for menstruation again. So ಹೇಗಾಗುತ್ತೆ ಅಂದರೆ ಮತ್ತೆ ಸೈಕಲ್ ರಿಪೀಟ್ ಆಗುತ್ತೆ. So this cycle of changes in the female reproductive system during reproductive phase is called menstrual cycle. Coming to the next aspects called fertilization and embryonic development. The process of fusion of male and female gametes is called fertilization, and the product of fertilization is called zygote. ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ, ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗಬೇಕಾದ್ರೆ ದರ್ ಆರ್ ಸರ್ಟನ್ ಈವೆಂಟ್ಸ್ ಆರ್ ರಿಕ್ವೈರ್ಡ್. ಈವೆಂಟ್ಸ್ ಯಾವುದು ಅಂತ ಹೇಳಿದ್ರೆ ಸ್ಪರ್ಮು ಕೂಡ ರಿಲೀಸ್ ಆಗಬೇಕಾಗುತ್ತೆ ಅದೇ ಟೈಮಲ್ಲಿ, ಮತ್ತು ಎಗ್ಗು ಕೂಡ ರಿಲೀಸ್ ಆಗಬೇಕಾಗುತ್ತೆ. ದಟ್ ಇನ್ಶೂಸ್ ಫರ್ಟಿಲೈಸೇಷನ್. ಸೊ ಡ್ಯೂರಿಂಗ್ ಫರ್ಟಿಲೈಸೇಷನ್ ಅಂದರೆ ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗ್ತಕ್ಕಂಥ ರೀಜನ್ ಯಾವುದು ಅಂತ ಗೊತ್ತಿದೆ. ಇಟ್ಸ್ ಅಪ್ಪರ್ ಲಿಂಬ್ ಆಫ್ ಫೆಲೋಪಿಯನ್ ಟ್ಯೂಬ್ ಆರ್ ಆಲ್ಸೋ ಕಾಲ್ಡ್ ಆಂಪ್ಲರಿ ಇಸ್ಥಮಿಕ್ ಜಂಕ್ಷನ್. ಸೊ ಫರ್ಟಿಲೈಸೇಷನ್ ಇಸ್ ದ ಪ್ರಾಸೆಸ್ ಆಫ್ ಫ್ಯೂಷನ್ ಆಫ್ ಮೇಲ್ ಅಂಡ್ ಫೀಮೇಲ್ ಗ್ಯಾಮಿಟ್ಸ್. During fertilization, the egg released by the ovary enters into fallopian tube and it waits for the sperm to reach and fertilize it. So during sexual intercourse, millions of sperms are released into vagina. So these sperms move towards the egg which is present in the fallopian tube with the help of tail, and also due to contractions of the uterus. As a result, so this egg is surrounded by millions of sperms in the fallopian tube. So of these, only one sperm is capable of fertilizing the egg and the rest degenerates. Now, so sperm enters into the cytoplasm of the egg through zona pellucida and plasma membrane. The entry of the egg into cytoplasm activates the egg. This is called activation of egg. As a result, the membranes of the egg gets thickened ದಿಸ್ ಇಸ್ ಟು ಎನ್ಶೂರ್ ಮೋನೋಸ್ಪರ್ಮಿ. ಇಲ್ಲಿ ಒಂದೇ ಸ್ಪರ್ಮ್ ಎಂಟ್ರಿ ಆಗಬೇಕು, ಯಾಕಂದ್ರೆ ಅದನ್ನೇ ನಾವು ಮೋನೋಸ್ಪರ್ಮಿ ಅಂತ ಕರಿತೀವಿ. ದ ಎಂಟ್ರಿ ಆಫ್ ಎ ಸಿಂಗಲ್ sperm into the egg is called monospermy. On the contrary, polyspermy is the entry of more than one sperm in some other organisms. But human beings only, it is to ensure monospermy, and to ensure monospermy, the membranes of the egg gets thickened. ಏನಾಯಿತು, ಸ್ಪರ್ಮ್ ಈಗ ಇಟ್ ಎಂಟರ್ಸ್ ಇನ್ ಟು ದ ಸೈಟೋಪ್ಲಾಸಮ್ ಆಫ್ ದ ಎಗ್. ಇಲ್ಲಿ ಯಾವ ಪಾರ್ಟ್ ಎಂಟ್ರಿ ಆಗುತ್ತೆ ಅಂದ್ರೆ ಓನ್ಲಿ ದ ಸ್ಪರ್ಮ್ ಹೆಡ್ ಕ್ಯಾನ್ ಗೇನ್ ಎಂಟ್ರಿ ಟು ದ ಸೈಟೋಪ್ಲಾಸಮ್, ಮಿಡಲ್ ಪೀಸ್ as well as tail remains outside. As soon as the sperm head enters into the cytoplasm, it gets activated. The secondary oocyte, it has completed only meiosis 1, starts completing meiosis 2. As a result, secondary polar bodies are extruded at this stage. ಅಂದ್ರೆ ಆ ಸೆಕೆಂಡರಿ ಪೋಲಾರ್ ಬಾಡೀಸ್ ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ ಈ ಪೆರಿವಿಟಲೈನ್ ಸ್ಪೇಸ್ಗೆ ಬರುತ್ತೆ, ಅವಾಗ ಫರ್ಟಿಲೈಸೇಷನ್ ಪ್ರಾಸೆಸ್ ಇನಿಷಿಯೇಷನ್ ಆಗುತ್ತೆ. ಇಲ್ಲಿ ದ ನ್ಯೂಕ್ಲಿಯಸ್ ಆಫ್ ದ sperm head is called male pronucleus and the nucleus of the egg is called female pronucleus. So fused to form a structure called zygote. So that is how the product of fertilization called zygote is formed. So during fertilization itself. What is the next process? ನಾನು ಹೇಳ್ತಾ ಇದ್ದೆ, fertilized egg undergo repeated mitotic divisions. ಒಂದು ಮೈಟೋಸಿಸ್ ಡಿವಿಷನ್ ಆದ್ರೆ ಆಗ್ತಕ್ಕಂಥ ಒಂದು ಪ್ರಾಸೆಸ್ಗೆ ನಾವೇಂತ ಕರಿತೀವಿ ಅಂದ್ರೆ ಎಂಬ್ರಿಯೋಜೆನಿಸಿಸ್ ಅಂತ ಕರಿತೀವಿ. ಎಂಬ್ರಿಯೋಜೆನಿಸಿಸ್ ಅಂತ ಹೇಳಿದ್ರೆ ದ ಪ್ರಾಸೆಸ್ ಫಾರ್ಮೇಶನ್ ಆಫ್ ಎಂಬ್ರಿಯೋ ಫ್ರಮ್ ಫರ್ಟಿಲೈಸ್ಡ್ zygote. Zygote undergoes repeated mitotic division. The repeated mitotic divisions of the zygote is called cleavage. The resulting cells are called as blastomers. ಈಗ ಕ್ಲೀವೇಜ್ ಆದ್ರೆ ಬರ್ತಕ್ಕಂಥ ಸೆಲ್ಸ್ಗೆ ನಾವು ಬ್ಲಾಸ್ಟೋಮರ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಈಗ ಇಲ್ಲಿ ನೋಡ್ತಾ ಇರಬಹುದು, ಈ ಒಂದು ಕಂಡೀಷನ್ಸ್ ಅಲ್ಲಿ 2 ಬ್ಲಾಸ್ಟೋಮರ್ಸ್ ಇದೆ, ಇಲ್ಲಿ 4 ಬ್ಲಾಸ್ಟೋಮರ್ಸ್ ಇದೆ, 8 ಬ್ಲಾಸ್ಟೋಮರ್ಸ್ ಇದೆ, ಹಾಗೆ 8 ಇಂದ 16, ಹಾಗೆ 32, ಹಾಗೆ 64, ಈ ರೀತಿ ಬ್ಲಾಸ್ಟೋಮರ್ಸ್ ಫಾರ್ಮೇಶನ್ ಆಗ್ತಾನೆ ಇರುತ್ತೆ. ಅಪ್ ಟು ಹಿಯರ್ ದ cleavage patterns are regular. ಚೆನ್ನಾಗಿ ನೋಡಬಹುದು. Later the cleavage patterns are irregular. The embryo between 16 to 32 cell stage is also known as morula. ಇದನ್ನ ಮಾರುಲಾ ಅಂತ ಕರಿತೀವಿ because it resembles the shape of a mulberry fruit. ಅಮೋರಸ್ ಆಲ್ಬಾ, ರೇಷ್ಮೆ ಹಣ್ಣು ಹೇಗಿದೆಯೋ ಅದೇ ರೀತಿ ಕಾಣೋದ್ರಿಂದ ಇದನ್ನ ಮಾರುಲಾ ಅಂತ ಕರಿತೀವಿ. ಸೊ ಮಾರುಲಾ is the first stage of embryonic development. ಈಗ ಮಾರುಲಾ ನೆಕ್ಸ್ಟ್ ಫರ್ದರ್ ಡೆವಲಪ್ಮೆಂಟ್ ಹೋಗುತ್ತೆ, ಅದನ್ನ ನಾವು ಬ್ಲಾಸ್ಟುಲಾ ಅಂತ ಕರಿತೀವಿ. Morula is a solid ball of blastomeres. So later ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ it develops a cavity. ಆ ಕ್ಯಾವಿಟಿ ನಾವು ಎಂತ ಕರಿತೀವಿ ಅಂದ್ರೆ ಬ್ಲಾಸ್ಟೋಸೀಲ್ ಅಂತ ಕರಿತೀವಿ. With the development of blastocele, this morula is transformed into blastula or blastocyst. ಈಗ ಒಂದು ಬ್ಲಾಸ್ಟೋಸೀಲ್ ಆಗಿ ಬ್ಲಾಸ್ಟೋಸಿಸ್ಟ್ ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗುತ್ತೆ. So this is the second embryonic stage in human embryo. And the embryonic development ಅಲ್ಲಿ blastocyst is the second stage. ಇಲ್ಲಿ ನೋಡಿ, ಈಗ ಫರ್ಟಿಲೈಸ್ಡ್ zygote undergoes ಕ್ಲೀವೇಜ್ ಆದ್ರೆ ಬರ್ತಕ್ಕಂಥ ಒಂದು ಸಾಲಿಡ್ ಬಾಲ್ ಆಫ್ ಬ್ಲಾಸ್ಟೋಮರ್ಸ್ಗೆ ನಾವು ಮಾರುಲಾ ಅಂತ ಕರಿತೀವಿ. ಸೊ ಮಾರುಲಾದಲ್ಲಿ ಏನಕ್ಕೆ ಡೆವಲಪ್ ಆಗುತ್ತೆ ಅಂದ್ರೆ ಒಂದು ಕ್ಯಾವಿಟಿ ಡೆವಲಪ್ ಆಗುತ್ತೆ, ಅದನ್ನ ಬ್ಲಾಸ್ಟೋಸೀಲ್ ಅಂತ ಕರಿತೀವಿ. ವಿತ್ ದ ಡೆವಲಪ್ಮೆಂಟ್ ಆಫ್ ಬ್ಲಾಸ್ಟೋಸೀಲ್ ದ ಮಾರುಲಾ ಈಸ್ ಸೆಟ್ ಟು ಬಿ ಬ್ಲಾಸ್ಟೋಸಿಸ್ಟ್ ಅಂತ ಕರಿತೀವಿ. ಇಲ್ಲಿ ಸ್ಪೇಸ್ ಇದೆ, ಕ್ಯಾವಿಟಿ ಇದೆ, ಬಿಕಾಸ್ ಆಫ್ ದಟ್ ಇಟ್ಸ್ ಆಲ್ಸೋ ನೋನ್ ಆಸ್ ಹಾಲೋ ಬಾಲ್ ಆಫ್ ಬ್ಲಾಸ್ಟೋಮರ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಇಟ್ ಈಸ್ ದ ಸೆಕೆಂಡ್ ಸ್ಟೇಜ್ ಆಫ್ ಎಂಬ್ರಿಯಾನಿಕ್ ಡೆವಲಪ್ಮೆಂಟ್ ಇನ್ ಹ್ಯೂಮನ್ ಬೀಯಿಂಗ್ಸ್. ನಾವು ಬ್ಲಾಸ್ಟುಲಾ ಮೂವ್ ಟುವರ್ಡ್ಸ್ ದ ಯುಟ್ರಸ್. ಇಲ್ಲಿ ಈ ಒಂದು ಫೆಲೋಪಿಯನ್ ಟ್ಯೂಬ್ ಇಲ್ಲಿಂದ ಏನು ಬ್ಲಾಸ್ಟುಲಾ ಇದೆ, ಇಟ್ ಮೂವ್ಸ್ ಟುವರ್ಡ್ಸ್ ದ ಯುಟ್ರಸ್ and it attaches to the wall of the endometrium. Endometrium ಅಂದ್ರೆ ಗೊತ್ತಿದೆ. The inner lining of the uterus is called endometrium, and thereby blastocyst attaches to the wall of the endometrium. So this process is called implantation of embryo. ಇಲ್ಲಿ ತೋರಿಸಿದೆ ನೋಡಿ. So this is the uterine epithelium ಅಥವಾ ಇಲ್ಲಿ ಎಂಡೋಮೆಟ್ರಿಯಂ ಇರುತ್ತೆ. ಈ ಒಂದು ರೀಜನ್ಸ್ಗೆ ಬ್ಲಾಸ್ಟೋಸಿಸ್ ಅಟ್ಯಾಚ್ ಆಗುತ್ತೆ ಅಂತ ಒಂದು ಕಂಡೀಷನ್ಸ್ಗೆ ನಾವು ಇಂಪ್ಲಾಂಟೇಷನ್ ಆಫ್ ಎಂಬ್ರಿಯೋ ಅಂತ ಕರಿತೀವಿ. So this leads to pregnancy. Coming to the structure of human blastocyst. Human blastocyst is a stage of embryonic development. First stage ನಾವು ಮಾರುಲಾ ಅಂತ ಕರಿತೀವಿ. ಮಾರುಲಾ develops into blastula. In human beings, it is said to be blastocyst ಅಂತ ಕರಿತೀವಿ. It's spherical in shape, covered over by outermost layer of cells. ಅದನ್ನು ನಾವು ಟ್ರೋಪೋಬ್ಲಾಸ್ಟ್ ಅಂತ ಕರಿತೀವಿ, ಇಲ್ಲ ಟ್ರೋಪೋಎಕ್ಟೋಡರ್ಮ್ ಅಂತ ಕರಿತೀವಿ. It encloses a inner cell mass. ಇದಕ್ಕೆ ನಾವು embryoblast ಅಂತ ಕರಿತೀವಿ. So the cavity present in the blastocyst is called as blastocele. It's filled with blastocelic fluid secreted by surrounding blastomeres. ಹಾಗಾದ್ರೆ what is the fate of these cells? ಇಲ್ಲಿ ಎರಡು ಲೇಯರ್ಸ್ ನಾವು ಕಾಮನ್ ಆಗಿ ಡಿಸ್ಟಿಂಗ್ವಿಶ್ ಮಾಡಬಹುದು. outermost layer of cells surrounding the blastocyst is tropoblast, and inner cell mass ಗೆ ನಾವು embryoblast ಅಂತ ಕರಿತೀವಿ. The tropoblastic cells which are in contact with the embryoblast or embryonic knob is said to be cells of Rober. ಹಾಗಾದ್ರೆ ಏನಾಗಬಹುದು ಟ್ರೋಪೋಬ್ಲಾಸ್ಟ್ ಅಥವಾ ಟ್ರೋಪೋ ಎಕ್ಟೋಡರ್ಮ್ ಏನಾಗಬಹುದು ಅಥವಾ ಇನ್ನರ್ ಸೆಲ್ ಮಾಸ್ ಏನಾಗಬಹುದು ಅಂದ್ರೆ ಟ್ರೋಪೋಬ್ಲಾಸ್ಟ್ ಆರ್ ಟ್ರೋಪೋ ಎಕ್ಟೋಡರ್ಮ್ ಫಾರ್ಮ್ಸ್ ದ ಎಕ್ಸ್ಟ್ರಾ ಎಂಬ್ರಿಯಾನಿಕ್ ಮೆಂಬ್ರೇನ್ಸ್. ಇಲ್ಲಿ ಎಕ್ಸ್ಟ್ರಾ ಎಂಬ್ರಿಯಾನಿಕ್ ಮೆಂಬ್ರೇನ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಈಗ ಫೀಟಿಸ್ ಇಸ್ ಕವರ್ಡ್ ಬೈ 2 to 3 ಮೆಂಬ್ರೇನ್ಸ್. ಅವನ್ನೇ ನಾವು ಎಕ್ಸ್ಟ್ರಾ ಎಂಬ್ರಿಯಾನಿಕ್ ಮೆಂಬ್ರೇನ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಅದು ಕೋರಿಯಾನ್. ಓಕೆ, ಒನ್ ಆಫ್ ದ ಮೆಂಬ್ರೇನ್ ಇಸ್ ಕೋರಿಯಾನ್, ಆಮ್ನಿಯಾನ್, ಅಲಂಟೈಸ್ ಅಂಡ್ ಯೋಕ್ ಸಾಕ್. ದೀಸ್ ಆರ್ ದ 4 extra embryonic membranes of the developing embryo. ಅದ್ರ ಜೊತೆಗೆ ಟ್ರೋಪೋಬ್ಲಾಸ್ಟ್ or ಟ್ರೋಪೋಬಕ್ಟ್ ಏನ್ ಮಾಡುತ್ತೆ ಅಂದ್ರೆ ದ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ರೋಲ್ ಈಸ್ ಇಟ್ ಟೇಕ್ ಪಾರ್ಟ್ ಇನ್ ದ ಪ್ರಾಸೆಸ್ ಫಾರ್ಮೇಶನ್ ಆಫ್ ಪ್ಲಾಸೆಂಟಾ. ಪ್ಲಾಸೆಂಟಾ ಫಾರ್ಮೇಶನ್ ಕೂಡ ಈ ಒಂದು ಲೇಯರ್ ಇಂದ ಆಗುತ್ತೆ. ಟ್ರೋಪೋಬ್ಲಾಸ್ಟ್ ಅಥವಾ ಟ್ರೋಪೋ ಎಕ್ಟೋಡರ್ಮ್ ಇಟ್ಸ್ ಅ ಮೇಜರ್ ಪಾರ್ಟ್ ಫ್ರಮ್ ದ ಮೇಜರ್ ಪಾರ್ಟ್ ಡ್ಯೂರಿಂಗ್ ದ ಪ್ರಾಸೆಸ್ ಆಫ್ ಫಾರ್ಮೇಶನ್ ಆಫ್ ಪ್ಲಾಸೆಂಟಾ. ಹಾಗಾದ್ರೆ ಇನ್ನರ್ ಸೆಲ್ ಮಾಸ್ ಏನಾಗಬಹುದು, ಇಲ್ಲ ಟ್ರೋಪೋಬ್ಲಾಸ್ಟ್ ಏನಾಗಬಹುದು. ಇನ್ನರ್ ಸೆಲ್ ಮಾಸ್ ಆರ್ ಎಂಬ್ರಿಯೋಬ್ಲಾಸ್ಟ್ ಡೆವಲಪ್ಸ್ ಇನ್ ಟು embryo. ಹೀಗೆ ದಿಸ್ ಇಸ್ ದ ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ದ ಬ್ಲಾಸ್ಟೋಸಿಸ್ಟ್. ನೆಕ್ಸ್ಟ್ ಏನಾಗಬಹುದು, ಬ್ಲಾಸ್ಟುಲಾ ಇಸ್ ಕನ್ವರ್ಟ್ ಟು ಅನದರ್ ಸ್ಟ್ರಕ್ಚರ್ ಕಾಲ್ಡ್ ಗ್ಯಾಸ್ಟ್ರುಲಾ. ಈಗ ನಾವು 3 ಸ್ಟೇಜಸ್ಗೆ ಬಂದಿದ್ದೀವಿ. ದ ಫಸ್ಟ್ ಸ್ಟೇಜ್ ಇಸ್ ಕಾಲ್ಡ್ ಮಾರುಲಾ. ದ ಫಸ್ಟ್ ಸ್ಟೇಜ್ ಆಫ್ ಎಂಬ್ರಿಯಾನಿಕ್ ಡೆವಲಪ್ಮೆಂಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಮಾರುಲಾ. ಇಟ್ಸ್ ಅ ಸಾಲಿಡ್ ಬಾಲ್ ಆಫ್ ಬ್ಲಾಸ್ಟೋಮಿಯರ್ಸ್. ಹಾಗೆ ಸೆಕೆಂಡ್ ಸ್ಟೇಜ್ ಆಫ್ ಬ್ಲಾಸ್ಟುಲಾ ಇಸ್ ಕಾಲ್ಡ್ ಬ್ಲಾಸ್ಟುಲಾ ಅಥವಾ ಬ್ಲಾಸ್ಟೋಸಿಸ್ಟ್ ಅಂತ ಕರಿತೀವಿ. ಆ ಸೆಕೆಂಡ್ ಸ್ಟೇಜ್ ಆಫ್ ಎಂಬ್ರಿಯಾನಿಕ್ ಡೆವಲಪ್ಮೆಂಟ್ಗೆ ಬ್ಲಾಸ್ಟುಲಾ ಬ್ಲಾಸ್ಟೋಸಿಸ್ಟ್ ಅಂತ ಕರಿತೀವಿ. ಇಟ್ಸ್ ಎ hollow ball of blastomeres because of the presence of a cavity called blastocele. Now, blastula or blastocysts undergo further development. So, into gastrula, the process of formation of gastrula is called gastrulation. So, gastrulation involves the movement of blastomas in different directions. The movement of blastomas in different directions is also known as according to their fate. ಯಾಕಂತ ಹೇಳಿದ್ರೆ ಬ್ಲಾಸ್ಟೋಮಸ್ ಒಂದೊಂದು ಆರ್ಗನ್ ಅಥವಾ ಆರ್ಗನ್ ಸಿಸ್ಟಮ್ಸ್ ಏನಾಗ್ತವೆ ಫಾರ್ಮೇಶನ್ ಆಗೋದಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡ್ತವೆ. ದ ಮೂವ್ಮೆಂಟ್ ಆಫ್ ಬ್ಲಾಸ್ಟೋಮರ್ಸ್ ಇನ್ ಡಿಫರೆಂಟ್ ಡೈರೆಕ್ಷನ್ಸ್ ಅಕಾರ್ಡಿಂಗ್ ಟು ದೇರ್ ಫೇಟ್ ಆರ್ ಕಲೆಕ್ಟಿವ್ಲಿ ಕಾಲ್ಡ್ ಮಾರ್ಫೋಜೆನಿಕ್ ಮೂವ್ಮೆಂಟ್ಸ್. ಸೊ ಮಾರ್ಫೋಜೆನಿಕ್ ಮೂವ್ಮೆಂಟ್ results in the organization of all the blastomeres into 3 different layers. ಆ ಲೇಯರ್ಸ್ ಯಾವ್ದು ಅಂತ ಹೇಳಿದ್ರೆ the outermost layer is called ectoderm and the endoderm is the innermost layer and the third layer is called mesoderm. ಈ ಎಕ್ಟೋಡರ್ಮ್ ಮತ್ತೆ ಎಂಡೋಡರ್ಮ್ ಮಧ್ಯದಲ್ಲಿ ಬರ್ತಕ್ಕಂಥ ಒಂದು ಜರ್ಮ್ ಲೇಯರ್ಗೆ ನಾವು ಮೀಸೋಡರ್ಮ್ ಅಂತ ಕರಿತೀವಿ. ಈ ಎಕ್ಟೋಡರ್ಮ್, ಮೀಸೋಡರ್ಮ್ ಅಂಡ್ ಎಂಡೋಡರ್ಮ್ ಇವು ಕೂಡ ಪ್ರೈಮರಿ ಜರ್ಮ್ ಲೇಯರ್ಸ್. ಹಾಗೆನ ಕ್ಯಾವಿಟಿ ಕೂಡ ಇದೆ. ಆ ಕ್ಯಾವಿಟಿ ನಾವು ಕಾಮನ್ಗೆ ಗ್ಯಾಸ್ಟ್ರೋಸೀಲ್ ಅಂತ ಕರಿತೀವಿ, ಇಲ್ಲ ಆರ್ಕೆಂಟಿರಾನ್ ಅಂತ ಕರಿತೀವಿ. ಈಗ ಒಂದೊಂದರಲ್ಲಿ ಮಾರಲ್ದಲ್ಲಿ ಕ್ಯಾವಿಟಿ ಇರೋಲ್ಲ, ಸೊ ಇಟ್ಸ್ ಎ ಸಾಲಿಡ್ ಬಾಲ್ ಆಫ್ ಬ್ಲಾಸ್ಟೋಮಿಯರ್ಸ್. ಬ್ಲಾಸ್ಟೋದಲ್ಲಿ ಕ್ಯಾವಿಟಿ ಇರುತ್ತೆ, ಅದಕ್ಕೆ ಬ್ಲಾಸ್ಟೋಸಿಲ್ ಕ್ಯಾವಿಟಿ ಇರೋದ್ರಿಂದ ಬ್ಲಾಸ್ಟುಲಾ ಅಂತ ಕರಿತೀವಿ. ಅದೇ ರೀತಿ ಗ್ಯಾಸ್ಟ್ರೋಸಿಲ್ ಕ್ಯಾವಿಟಿ ಆರ್ ಆರ್ಕೆಂಟಿನಾ ಇರೋದ್ರಿಂದ ಇದನ್ನ ನಾವು ಗ್ಯಾಸ್ಟ್ರುಲಾ ಅಂತ ಕರಿತೀವಿ. ಸೊ ಎಕ್ಟೋಡರ್ಮ್, ಮೀಸೋಡರ್ಮ್ ಮತ್ತೆ ಎಂಡೋಡರ್ಮ್ ಇವು 3 ಸೇರಿಸಿ ನಾವು ಎಂತ ಕರಿತೀವಿ, ಪ್ರೈಮರಿ ಜರ್ಮ್ ಲೇಯರ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಹಾಗಾದ್ರೆ ವಾಟ್ ಇಸ್ ದ ಸಿಗ್ನಿಫಿಕೆನ್ಸ್ ಆಫ್ ದೀಸ್ ಪ್ರೈಮರಿ ಜರ್ಮ್ ಲೇಯರ್ಸ್? ಈ ಪ್ರೈಮರಿ ಜರ್ಮ್ ಲೇಯರ್ಸ್ ಸಿಗ್ನಿಫಿಕೆನ್ಸ್ ಏನಂತ ಹೇಳಿದ್ರೆ ಈಚ್ ಅಂಡ್ ಎವ್ರಿ ಪ್ರೈಮರಿ ಜರ್ಮ್ ಲೇಯರ್ ಗಿವ್ಸ್ ರೈಸ್ ಟು ಆರ್ಗನ್ಸ್ ಅಂಡ್ ಆರ್ಗನ್ ಸಿಸ್ಟಮ್ಸ್ ಆಫ್ ದ ಎಂಬ್ರಿಯೋ. ಹೇಗಾಗಬಹುದು ನೋಡಿರ್ಬೋದು ನಿಮ್ಗೆ ಇಲ್ಲಿ. ಸೊ ಎಕ್ಟೋಡರ್ಮ್ ಇದೆ, ಮೀಸೋಡರ್ಮ್ ಇದೆ, ಎಂಡೋಡರ್ಮ್ ಇದೆ. ಎಕ್ಟೋಡರ್ಮ್ ಗಿವ್ಸ್ ರೈಸ್ ಟು ಸ್ಕಿನ್ ಸೆಲ್ಸ್ ಆಸ್ ವೆಲ್ ಆಸ್ ನರ್ವಸ್ ಸೆಲ್ಸ್ ಅಥವಾ ನ್ಯೂರಾನ್ಸ್ಗೆ ಫಾರ್ಮೇಷನ್ ಆಗೋದಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡುತ್ತೆ. ಮೀಸೋಡರ್ಮ್ ಕಾರ್ಡಿಯಾಕ್ ಮಸಲ್ಸ್, ಸ್ಕೆಲೆಟಲ್ ಮಸಲ್ಸ್, ಹಾಗೆ ಟ್ಯೂಬ್ಯೂಲ್ ಸೆಲ್ ಆಫ್ ಕಿಡ್ನಿ, ರೆಡ್ ಬ್ಲಡ್ ಸೆಲ್ಸ್ ಆಗಿರ್ಬೋದು, ಸ್ಮೂತ್ ಮಸಲ್ ಸೆಲ್ಸ್ ಆಗಿರ್ಬೋದು ಫಾರ್ಮೇಷನ್ ಆಗೋದಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡುತ್ತೆ. ಹಾಗೆ ಎಕ್ಟೋಡರ್ಮ್ ಲಂಗ್ ಸೆಲ್ಸ್ ಅಂದ್ರೆ ಪಾರ್ಟ್ಸ್ ಆಫ್ ರೆಸ್ಪಿರೇಟರಿ ಸಿಸ್ಟಮ್, ಹಾಗೆ ಡೈಜೆಸ್ಟಿವ್ ಸಿಸ್ಟಮ್, ಥೈರಾಯ್ಡ್ ಸೆಲ್, ಹಾಗೆ ಪ್ಯಾಂಕ್ರಿಯಾಟಿಕ್ ಸೆಲ್ಸ್. ಅಂದ್ರೆ ಈಚ್ ಅಂಡ್ ಎವ್ರಿ ಜರ್ಮ್ ಲೇಯರ್ ಈಸ್ ರೆಸ್ಪಾನ್ಸ್ ಅಂದ್ರೆ 3 ಜರ್ಮ್ ಲೇಯರ್ ಇರುತ್ತೆ, ಎಕ್ಟೋಡರ್ಮ್, ಎಂಡೋಡರ್ಮ್ and the middle one is called mesoderm. They give rise to specific organs and organs of the embryo. The process of formation of organs and organs of the organs is also known as organogenesis. ಹೇಗೆ ಡಿಫೈನ್ ಮಾಡ್ತೀವಿ? The process of formation of organs from these primary germ layers is called organogenesis. ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ major organs are formed by 3 months of pregnancy. Human placenta. The very important stage in the development of human embryo is formation of placenta. So placenta formation is a vital stage in the embryonic development in case of human beings. Why placenta? ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ ಮಾರುಲಾ develops into blastula, blastula develops into gastrula and undergo further development. The developing embryo demands more nutrition from the mother's side. ಅದಕ್ಕೋಸ್ಕರ mother establishes a contact or embryonic connection between the fetus. The embryonic connection that is established between the fetus and the uterine wall is called placenta. So therefore placenta can be defined as an embryonic connection between the fetus and uterus for the purpose of physiological exchange of materials. Even the structure placenta anta karitivi. So placenta is formed both the tissues of the fetus as well as mother. The fetal part of the placenta is chorion as well as allantois. ಈಗ ಪ್ಲಾಸೆಂಟಾ ಪಾರ್ಟ್ ಅಲ್ಲಿ ಯಾವ್ದು ಬರುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ ಕೋರಿಯಾನ್ ಬರುತ್ತೆ ಮತ್ತೆ as well as allantois ಬರುತ್ತೆ. ಆ ಎರಡು ಕೂಡ ಈ ಪ್ಲಾಸೆಂಟಸ್ ಅದು ಎಂಬ್ರಿಯೋ ಸೈಡ್ ಇಂದ ಅಥವಾ ಫೀಟಸ್ ಸೈಡ್ ಇಂದ ಪ್ಲಾಸೆಂಟಾ ಫಾರ್ಮೇಷನ್ ಆಗೋದಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡುತ್ತೆ. ಅಂಡ್ ದ ಮೆಟರ್ನಲ್ ಪಾರ್ಟ್ ಆಫ್ ದ ಪ್ಲಾಸೆಂಟಾ ಇಸ್ ಕಾಲ್ಡ್ ಯುಟ್ರೈನ್ ಎಪಿಥೀಲಿಯಂ. ಇಲ್ಲಿ ನೋಡ್ತಾ ಇರಬಹುದು ದಿಸ್ ಔಟರ್ ಮೋಸ್ಟ್ ಲೇಯರ್ ಆಫ್ ದ embryo. It forms a finger-like projections. So these finger-like projections are called as chorionic villi. So these chorionic villi ಏನಾಗುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ invade the endometrium of the mother till they reach blood vessels. As a result, so these chorionic villi are dipped in the maternal blood. Such a placenta is called hemochorial placenta. ಯಾಕಂದ್ರೆ ಹ್ಯೂಮನ್ ಪ್ಲಾಸೆಂಟಾ ಹಿಮೋಕೋರಿಯಲ್ ಪ್ಲಾಸೆಂಟಾ ಅಂತ ಕರಿತೀವಿ. ಯಾಕಂತ ಹೇಳಿದ್ರೆ ಈಗ ಇಲ್ಲಿ ಎಂಬ್ರಿಯೋ is surrounded by chorion, its outermost fetal membrane ಅಂತ ಕರಿತೀವಿ. It forms a finger-like projections. ಈ projectionsಗೆ ನಾವು chorionic villi ಅಂತ ಕರಿತೀವಿ. ಈ chorionic villi ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ ಸೊ ಎಂಟರ್ಸ್ ಇಂಟು ಯುಟ್ರೈನ್ ಎಪಿಥೀಲಿಯಂ, ಒಳಗಡೆ ಹೋಗುತ್ತೆ. ಎಲ್ಲಿವರೆಗೂ ಹೋಗುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ ಆ ಬ್ಲಡ್ ವೆಲ್ಸ್ ರೀಚ್ ಆಗೋವರೆಗೂ ಹೋಗುತ್ತೆ. ಸೊ ಆಸ್ ಅ ರಿಸಲ್ಟ್ chorionic villi ಆರ್ ಡಿಪ್ಡ್ ಇನ್ ದ ಮೆಟರ್ನಲ್ ಬ್ಲಡ್. ಅದಕ್ಕೋಸ್ಕರ ಇಂಥ ಒಂದು ಪ್ಲಾಸೆಂಟಾಗಿ ನಾವು ಎಂತ ಕರಿತೀವಿ ಅಂದ್ರೆ ಹೀಮೋಕೋರಿಯಲ್, ಹೀಮೋ ಅಂದ್ರೆ ಬ್ಲಡ್ ಅಂತ. ಸೊ ಇಲ್ಲಿ chorionic villi ಇರೋದ್ರಿಂದ ಹೀಮೋಕೋರಿಯಲ್ ಪ್ಲಾಸೆಂಟಾ ಅಂತ ಕರಿತೀವಿ. ಇಲ್ಲಿ ದ ಅದರ್ ನೇಚರ್ ಆಫ್ ದಿಸ್ ಪ್ಲಾಸೆಂಟಾ ಇಸ್ ಕೋರಿಯೋ-ಅಲಂಟಾಯಿಕ್. ಯಾಕಂತ ಹೇಳಿದ್ರೆ ದ ಮೇಜರ್ ಪಾರ್ಟ್ ಆಫ್ ಪ್ಲಾಸೆಂಟಾ ಇಸ್ ಫ್ರಮ್ ಫೀಟಲ್ ಸೈಡ್. ಯಾವ್ದಂತ ಹೇಳಿದ್ರೆ ಸೊ ಬೋತ್ ಕೋರಿಯಾನ್ ಆಸ್ ವೆಲ್ ಆಸ್ ಅಲಂಟಾಯ್ಸ್ ಟೇಕ್ ಪಾರ್ಟ್ ಇನ್ ಪ್ಲಾಸೆಂಟಾ ಫಾರ್ಮೇಷನ್. ಈ ಕೋರಿಯಾನ್ ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ ಇಟ್ ಫಾರ್ಮ್ಸ್ chorionic villi and the blood vessels produced by allantois extended into it. ಅದಕ್ಕೆ ಹ್ಯೂಮನ್ ಪ್ಲಾಸೆಂಟಾ ನಾವೇಂತ ಕರಿತೀವಿ ಅಂದ್ರೆ ಕೋರಿಯೋ-ಅಲಂಟಾಯಿಕ್ ಪ್ಲಾಸೆಂಟಾ ಅಂತ ಕರಿತೀವಿ. ಸೊ ಅದರ್ ಕಂಡೀಷನ್ ಏನಂತ ಹೇಳಿದ್ರೆ ಡೆಸಿಡಿಯೇಟ್. ಅಟ್ ದ ಟೈಮ್ ಆಫ್ ಪಾರ್ಚುರೇಷನ್ ಆರ್ ಚೈಲ್ಡ್ ಬರ್ತ್ ದ ಪ್ಲಾಸೆಂಟಾ ಆಟೋಮೆಟಿಕಲಿ ಸಪರೇಟ್ಸ್. ಇಂಥ ಒಂದು ಕಂಡೀಷನ್ಸ್ಗೆ ನಾವು ಡೆಸಿಡಿಯೇಟ್ ಅಂತ ಕರಿತೀವಿ. ಸೊ ದಿಸ್ ಇಸ್ ದ ನೇಚರ್ ಆಫ್ ಹ್ಯೂಮನ್ ಪ್ಲಾಸೆಂಟಾ. ಇಟ್ ಈಸ್ ಹಿಮೋಕೋರಿಯಲ್, ಇಟ್ ಈಸ್ ಕೋರಿಯೋ-ಅಲಂಟಾಯಿಕ್ as well as deciduate. And at the same time, so this is a structure called placenta. This structure is supported by umbilical cord. A cord-like structure around the placenta is called umbilical cord. So umbilical cord carries 3 veins, ಅದರಲ್ಲಿ 2 arteries ಅಂತ ಕರಿತೀವಿ and 1 is called vein. Arteries function ಏನಂತ ಹೇಳಿದ್ರೆ they carry deoxygenated blood from the fetus to mother, whereas veins carry oxygenated blood from the mother to the fetus. So this is the significance of umbilical cord. So ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ, so placenta acts as a temporary endocrine gland and it secretes various hormones. ಅದರಲ್ಲಿ ಮುಖ್ಯವಾಗಿ HCG ಆಗ್ಬೋದು, human chorionic gonadotropins, human placental lactogen as well as progestogens ಮತ್ತೆ lactogens. ಸೊ ಇವನ್ನ ನಾವು ಕಾಮನ್ಗೆ ಹಾರ್ಮೋನ್ ಸೆಕ್ರೆಟೆಡ್ ಬೈ ಪ್ಲಾಸೆಂಟಾ ಅಂತ ಕರಿತೀವಿ. The total time required for the complete development of embryo is called gestation period. After gestation period, the fetus will be expelled out of the uterus. The process is called parturition. Parturition is a complex neuroendocrine mechanism. The signals for parturition originate from placenta and fetus. Induce mild uterine contractions. These are called fetal ejection reflex. Oxytocin secreted by posterior lobe of pituitary plays a vital role as far fetal ejection reflex is concerned during childbirth or parturition. After parturition, mother starts producing milk in the mammary gland. The process of production of milk in the mammary glands following parturition is called lactation. The first milk secreted in the initial few days of lactation is called colostrum. It is rich in IgA antibodies. So that's the reason why breastfeeding is always recommended by doctors. So that's synapsis part. Synapsis part ಆದಮೇಲೆ okay, some model questions ಈ ಒಂದು ಚಾಪ್ಟರ್ಲಿ ನಾವು ಡಿಸ್ಕಸ್ ಮಾಡೋಣ. Look at the first question. Primary sex organs refers to. So ಇಲ್ಲಿ ನಾನು ಫಸ್ಟ್ ಸಿನಾಪ್ಸಿಸ್ ಹೇಳಬೇಕಾದ್ರೆ ಹೇಳಿದ್ದೀನಿ. Sex organs that produce gametes, this is a function of a primary sex organ. Sex organs that transport gametes, this is a function of a secondary sex organ. So it does not belong to this question. Then sex organs that secrete sex hormones is a function of primary sex organs. Since both the options are there in D, so therefore D is the right option. Primary sex organs refers to sex organs that produce gametes and secretes hormones. Location of Leydig cells and their secretions are. It's a direct question. It's a concept based question. ಅದು ಟೆಸ್ಟಿಸ್ ಮೇಲೆ ಬೇಸ್ ಆಗಿದೆ. Leydig cells are interstitial cells present in between the seminiferous tubules. So ಅವು ಬರುತ್ತೆ ಅಂದರೆ testis ಅಲ್ಲಿ ಬರುತ್ತೆ. They meant for secretion of a male sex hormone called testosterone. So therefore the right option is B. Look at the next question. Which of the following is not a male accessory gland? ಹೇಳಿದ್ದೀನಿ synapsis spot time ಅಲ್ಲಿ. Accessory glands of the male reproductive system includes a pair of seminal vesicles, a prostate gland, and a pair of corpus glands also called bulbourethral glands. ಇಲ್ಲಿ vestibular glands ಬರೋದು female reproductive system ಅಲ್ಲಿ. So therefore, so this is not the option. ಅಂದರೆ ಈ ಒಂದು ಆಪ್ಷನ್ ಇದಕ್ಕೆ ಬರೋದಿಲ್ಲ. So therefore, the right option is D. Next question ನೋಡಿ. The seminiferous tubules open into a network called. All the seminiferous tubules of a testis opens into a network called retitestis, which in turn open into vasa efferentia. Vasa efferentia open into epididymis. Epididymis opens into vasa deferens. ಈ ಕ್ವಶನ್ ಅಲ್ಲಿ the seminiferous tubules open into a network. ಅದಕ್ಕೆ ಕರೆಕ್ಟ್ ಆಪ್ಷನ್ ಯಾವುದು ಅಂತ ಹೇಳಿದ್ರೆ retitestis. A is the right option. Coming to the next question, identify the correct route for passage of sperms from testis to exterior in male reproductive system. ಗೊತ್ತಿದೆ seminiferous tubules where spermatogenesis takes place. Sperms from seminiferous tubules enter into retitestis. ಈ ಒಂದು ಪ್ರಾಸೆಸ್ಗೆ ನಾವು ಕಾಮನ್ ಆಗಿ ಸ್ಪರ್ಮಿಯೇಷನ್ ಅಂತ ಕರಿತೀವಿ. ರೆಟಿಟೆಸ್ಟಿಸ್ ಇಂದ ಸ್ಪರ್ಮ್ ವ್ಯಾಸ ಎಫರೆನ್ಷಿಯಾ ಹೋಗಬೇಕು. ವ್ಯಾಸ ಎಫರೆನ್ಸ್ ಇಂದ ಎಪಿಡೈಡಮಸ್ ಬರುತ್ತೆ. ಎಪಿಡೈಡಮಸ್ ಇಂದ ವ್ಯಾಸ ಡಿಫರೆನ್ಸ್ಗೆ ಹೋಗುತ್ತೆ. ದೆನ್ ವ್ಯಾಸ ಡಿಫರೆನ್ಸ್ ಇಂದ ಇಸಾಕ್ಯುಲರ್ ಡಕ್ಟ್ ಅಂಡ್ ಓಪನ್ಸ್ ಔಟ್ಸೈಡ್ ಥ್ರೂ ಯುರೇಥ್ರಾ. ಸೊ ದೇರ್ಫೋರ್ ದ ರೈಟ್ ಆಪ್ಷನ್ ಈಸ್ ಎ and the rest are wrong. Esaculate duct is formed by ಹೇಳ್ತಾ ಇದೆ synapsis time ಅಲ್ಲಿ. So esaculate duct ಅಂದ್ರೆ ಇದು ಫಾರ್ಮೇಷನ್ ಆಗೋದು ವ್ಯಾಸ ಡಿಫರೆನ್ಸ್ ಇಂದ. ವ್ಯಾಸ ಡಿಫರೆನ್ಸ್ of both the sides enter into abdomen, loop over the urinary bladder where they receive ducts from seminal vesicles and form a duct. ಈ ಒಂದು duct ಗೆ ನಾವು esaculate duct ಅಂತ ಕರಿತೀವಿ. Formed by seminal vesicles as well as vas difference. ಈ ಎರಡು ಆಪ್ಷನ್ಸ್ ಡಿ ನಲ್ಲಿ ಇರೋದ್ರಿಂದ ಸೊ ದಿಸ್ ದ ರೈಟ್ ಆಪ್ಷನ್ ಡಿ. ದ ಟ್ರಾನ್ಸ್ಫಾರ್ಮೇಷನ್ ಆಫ್ ಸ್ಪರ್ಮ್ಯಾಟಿಡ್ಸ್ ಇಂಟು ಸ್ಪರ್ಮ್ಸ್ ಇಸ್ ಟರ್ಮ್ಡ್ ಆಸ್. ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ ಸೊ ಸ್ಪರ್ಮ್ಯಾಟಿಡ್ಸ್ ಆರ್ ದ ರಿಸಲ್ಟ್ ಆಫ್ ಸೆಕೆಂಡ್ ಮಿಯಾಸಿಸ್ ಡ್ಯೂರಿಂಗ್ ಸ್ಪರ್ಮೆಟೋಜೆನಿಸಿಸ್. ಈ ಸ್ಪರ್ಮ್ಯಾಟಿಡ್ಸ್ ಆರ್ ನಾನ್ ಫಂಕ್ಷನಲ್. ಸೊ ದೀಸ್ ನಾನ್ ಫಂಕ್ಷನಲ್ ಸ್ಪರ್ಮ್ಯಾಟಿಡ್ಸ್ ಆರ್ ಟ್ರಾನ್ಸ್ಫಾರ್ಮ್ಡ್ ಇಂಟು ಸ್ಪರ್ಮ್ಸ್ ಬೈ ಅ ಪ್ರಾಸೆಸ್ ಕಾಲ್ಡ್ ಸ್ಪರ್ಮಿಯೋಜೆನಿಸಸ್. ಹಾಗಾದ್ರೆ ಸ್ಪರ್ಮೆಟೋಜೆನಿಸಿಸ್ ಅಂದ್ರೆ ದ ಪ್ರಾಸೆಸ್ ಆಫ್ ಫಾರ್ಮೇಷನ್ ಆಫ್ ಸ್ಪರ್ಮ್. ಸ್ಪರ್ಮೇಷನ್ ಅಂದ್ರೆ the release of sperms from seminiferous tubules into duct system. Hage insemination is nothing but the process of release of sperms into vagina during sexual intercourse. The question is the transformation of spermatids into sperms. So therefore the correct option is spermiogenesis. Look at the next question. The process of release of sperm from Sertoli cells of seminiferous tubules into duct system is termed as. It is the process called spermiation. ಈಗ ನಾನು ಹೇಳಿದ್ದೀನಿ ಲಾಸ್ಟ್ ಕ್ವಶನ್ ಅಲ್ಲಿ. ಸ್ಪರ್ಮೆಟೋಜೆನಿಸಿಸ್ ಇಸ್ ದ ಪ್ರಾಸೆಸ್ ಫಾರ್ಮೇಷನ್ ಆಫ್ ಸ್ಪರ್ಮ್ಸ್. ಸ್ಪರ್ಮಿಯೋಜೆನಿಸಿಸ್ ಇಸ್ ಎ ಡಿಫ್ರೆನ್ಸಿಯೇಷನ್ ಪ್ರಾಸೆಸ್. ಇಲ್ಲಿ ಅಂದ್ರೆ ಟ್ರಾನ್ಸ್ಫಾರ್ಮೇಷನ್ ಆಫ್ ಸ್ಪರ್ಮ್ಯಾಟಿಡ್ಸ್ ಇನ್ ಸ್ಪರ್ಮ್. ಇಲ್ಲಿ ಇನ್ನೊಂದು ಟರ್ಮ್ ಇದೆ ಸ್ಪರ್ಮ್ಯಾಟೋಇಲಿಯೋಸಿಸ್ ಅಂತ. ಸ್ಪರ್ಮಿಯೋಜೆನಿಸಿಸ್ ಮತ್ತೆ ಸ್ಪರ್ಮ್ಯಾಟೋಇಲಿಯೋಸಿಸ್ ಎರಡೂ ಕೂಡ ಒಂದೇ. ದೇ ಬೋತ್ ಆರ್ ಸೇಮ್. ಸೊ ದ meant for transformation of spermatids into sperms. So here the correct option is C. The cavity present in the Graafian follicle is. Graafian follicle structure ಹೇಳ್ತಾ ಇದೆ. ಸೊ ದಿಸ್ ಇಸ್ ಕಾಲ್ಡ್ ತೀಕ ಎಕ್ಸ್ಟರ್ನ್ ಅಂತ ಕರಿತೀವಿ. ಇದನ್ನೇ ತೀಕ ಇಂಟರ್ನ್ ಅಂತ ಕರಿತೀವಿ. ಸೊ ದಿಸ್ ಇಸ್ ಕಾಲ್ಡ್ ಮೆಮ್ಬ್ರೇನಾ ಗ್ರ್ಯಾನ್ಯುಲೋಸಾ. ಇದನ್ನ ಕ್ಯೂಮುಲಸ್ ಯೂಪೋರನ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ದಿಸ್ ಎನ್ಕ್ಲೋಸ್ ಸೆಕೆಂಡರಿ oocyte. The cavity present in it is called antrum. A direct question. So therefore the correct option is A. Seminal plasma. Seminal plasma is rich in fructose for providing nutrition or energy for sperms. It also composed of calcium as well as bicarbonate ions for providing alkaline medium. It also contains enzymes such as fibrinogens, prostaglandins, etc. So therefore, this is the right option and the rest are wrong. Coming to the next one, mammary duct is formed by the union of. Mammary breast structure study ಬೇಕಾದ್ರೆ the flow of milk ಹೇಗಾಗುತ್ತೆ ಅಂತ ಹೇಳ್ತಾ ಇದ್ದೆ. So mammary alveoli, these are milk secreting cells. They open into mammary tubules. Mammary tubules open into mammary duct. ಹೌದಾ? ಹಾಗಾದ್ರೆ the right option is ಯಾವುದು mammary tubules. ಈ mammary tubules ಜಾಯಿನ್ ಆದಾಗ mammary duct ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೆ. ಮ್ಯಾಮರಿ ಡಕ್ಟ್ ಫಾರ್ಮೇಷನ್ ಅಂದರೆ ಅದು ಓಪನ್ ಆಗೋದು ಮ್ಯಾಮರಿ ಆಂಪ್ಯುಲಾ ಅಂತ ಕರಿತೀವಿ. ಮ್ಯಾಮರಿ ಆಂಪ್ಯುಲಾ lactiferous duct open agi, it opens outside at the tip of the nipple. So therefore the correct option is mammary tubules. Identify the correct sequence of ejection of milk during breastfeeding the child by mother. ಹೀಗೆ ಹೇಳಿದೆ, ಮಿಲ್ಕ್ ಫ್ಲೋಸ್ ಫ್ರಮ್ ಮ್ಯಾಮರಿ ಆಲ್ವಿಯಲಿ ಇಂಟು ಮ್ಯಾಮರಿ ಟ್ಯೂಬ್ಯೂಲ್ಸ್. ಹಾಗಾದರೆ ಎ ಇಸ್ ದ ಮ್ಯಾಮರಿ ಆಲ್ವಿಯಲ್, ಅಲ್ಲಿಂದ ಎಲ್ಲಿಗೆ ಹೋಗಬೇಕು, ಬಿಗೆ ಹೋಗಬೇಕು. ಮ್ಯಾಮರಿ ಟ್ಯೂಬ್ಯೂಲ್ಸ್ ಓಪನ್ ಇಂಟು ಡಕ್ಟ್, ಹಾಗಾದರೆ ನೆಕ್ಸ್ಟು ಡಿಗೆ ಹೋಗಬೇಕು. ನೆಕ್ಸ್ಟು ಮ್ಯಾಮರಿ ಡಕ್ಟ್ಸ್ ಓಪನ್ ಇಂಟು ಮ್ಯಾಮರಿ ಆಂಪ್ಯುಲಾ, ನೆಕ್ಸ್ಟು ಸಿಗೆ ಹೋಗಬೇಕು. ನೆಕ್ಸ್ಟ್ ಮ್ಯಾಮರಿ ಆಂಪ್ಯುಲಾ ಓಪನ್ಸ್ ಇಂಟು ಲ್ಯಾಕ್ಟಿಫೆರಸ್ ಡಕ್ಟ್, ಅಂದರೆ ಎ ಬಿ ಡಿ ಸಿ ಇ ಈ ಆಪ್ಷನ್ಸ್ ಇರೋದು ಸಿನಲ್ಲಿ. ಸೊ ದೇರ್ಫೋರ್ ದ ರೈಟ್ ಆಪ್ಷನ್ ಈಸ್ ಸಿ. ಅಂದರೆ ಇದೇನು ತೋರಿಸು ಅಂದರೆ ಇಜೆಕ್ಷನ್, ತಾಯಿ ಮಗುಗೆ ಹಾಲು ಕುಡಿಸ್ಬೇಕಾದ್ರೆ ಆ ಹಾಲು ಹೋಗ್ತಕ್ಕಂಥ ರೀತಿ, ಅಂದರೆ ದ ಪ್ಯಾಸೇಜ್ ಅಥವಾ ಪ್ಯಾಸೇಜ್ ಆಫ್ ಮಿಲ್ಕ್ ಅಥವಾ ಇಜೆಕ್ಷನ್ ಆಫ್ ಮಿಲ್ಕ್ ಸೊ ಡ್ಯೂರಿಂಗ್ ಬ್ರೆಸ್ಟ್ ಫೀಡಿಂಗ್ ದ ಚೈಲ್ಡ್ ಬೈ ಮದರ್. Which is the correct sequence of layers in the egg from inside to outside? ನಾನು ಹೇಳ್ತಾ ಇದ್ದೆ. So this is the human egg which is surrounded by primary plasma membrane. ಹೌದಾ, ಪ್ಲಾಸ್ಮಾ ಮೆಂಬ್ರೇನ್ ಇರಬೇಕು ಈ ಒಂದರಲ್ಲಿ, ಯಾಕಂದರೆ ಇನ್ಸೈಡ್ ಟು ಔಟ್ಸೈಡ್ ಕೇಳಿರೋದ್ರಿಂದ ಪ್ಲಾಸ್ಮಾ ಮೆಂಬ್ರೇನ್ ಈ ಒಂದು ಆಪ್ಷನ್ಸ್ ಅಲ್ಲಿ ಇದೆ. ನೆಕ್ಸ್ಟ್ ಪ್ಲಾಸ್ಮಾ ಮೆಂಬ್ರೇನ್ ಇಸ್ ಸರೌಂಡೆಡ್ ಬೈ ಝೋನಾ ಪೆಲುಸಿಡಾ, a cellular or non-cellular layer. It's also a vitelline membrane which in turn covered by epithelium with radiating projections. Therefore, so this is the right option, that is B. Choose the correct option showing the events of human menstrual cycle in a proper order. ಇಲ್ಲಿ ಈವೆಂಟ್ ತೋರಿಸಿದ್ರೆ ಮೆನ್ಸ್ಟ್ರಲ್ ಸೈಕಲ್ ಅಲ್ಲಿ ದ ಫಸ್ಟ್ ಇವೆಂಟ್ ಏನಂತೆ ಸೆಕ್ರೇಷನ್ ಆಫ್ FSH ಅಂತ ಹೇಳಿದೆ, ಗ್ರೋತ್ ಆಫ್ ಕಾರ್ಪಸ್ ಲ್ಯೂಟಿಯಂ, ಡೆವಲಪ್ಮೆಂಟ್ ಆಫ್ ಮೆಚೂರ್ ಫಾಲಿಕಲ್, ಓವ್ಯುಲೇಷನ್, ಸಡನ್ ಇನ್ಕ್ರೀಸ್ ಇನ್ ದ ಲೆವೆಲ್ಸ್ ಆಫ್ LH. ಈಗ ಒಂದು ಮೆನ್ಸ್ಟ್ರಲ್ ಸೈಕಲ್ ನೋಡಿದ್ರೆ ನಿಮಗೆ ಐಡಿಯಾ ಬರುತ್ತೆ. ಇಲ್ಲಿ ಬಿಗಿನಿಂಗ್ ಅಲ್ಲಿ ಸೆಕ್ರೇಷನ್ ಆಫ್ ಫಾಲಿಕಲ್ ಸ್ಟಿಮ್ಯುಲೇಟಿಂಗ್ ಹಾರ್ಮೋನ್, ಅದೇನು ಮಾಡುತ್ತೆ, ಇಟ್ ಇಂಡ್ಯೂಸಸ್ ಫಾಲಿಕ್ಯುಲರ್ ಮೆಚುರೇಷನ್. ಆಸ್ ಅ ರಿಸಲ್ಟ್ ಏನಾಗಬಹುದು, ಗ್ರಾಫಿನ್ ಫಾಲಿಕಲ್ ಡೆವಲಪ್ ಆಗುತ್ತೆ. ಸೊ ಗ್ರಾಫಿನ್ ಫಾಲಿಕಲ್ ಡೆವಲಪ್ ಆದಾಗ ದೇರ್ ವಿಲ್ ಬಿ ಇನ್ಕ್ರೀಸ್ ಇನ್ ದ ಲೆವೆಲ್ಸ್ ಆಫ್ LH. ಅದೇನು ಮಾಡುತ್ತೆ, ಇಟ್ ಹೆಲ್ಪ್ಸ್ ಇನ್ ಎ ಪ್ರಾಸೆಸ್ ಕಾಲ್ಡ್ ಓವ್ಯುಲೇಷನ್. ಓವ್ಯುಲೇಷನ್ ಆದಮೇಲೆ ಈ ಒಂದು ಗ್ರಾಫಿನ್ ಫಾಲಿಕಲ್ ಏನಾಗಿ ಕನ್ವರ್ಟ್ ಆಗ್ಬೋದು, ಕಾರ್ಪಸ್ ಲ್ಯೂಟಿಯಂ ಆಗ್ಬೋದು. ದೇರ್ಫೋರ್ ಇಲ್ಲಿ ಆರ್ಡರ್ ಯಾವ್ದು ಬರುತ್ತೆ ಅಂದರೆ ಸೆಕ್ರೇಷನ್ ಆಫ್ FSH ಫಸ್ಟ್ ಎ ಬರುತ್ತೆ, ಆಮೇಲೆ ಡೆವಲಪ್ಮೆಂಟ್ ಆಫ್ ಏನು ಬರುತ್ತೆ, ಮೆಚೂರ್ ಫಾಲಿಕಲ್ ಬರುತ್ತೆ. ಎ ಇಸ್ ದ ಫಸ್ಟ್ ಒನ್, followed by that is C, development of mature follicle. Then mature follicle formation ಆದಮೇಲೆ increase in the levels of LH E ಬರುತ್ತೆ. E ಬಂದಮೇಲೆ LH ovulation ಆಗಬೇಕು under the influence of this LH, ಅದನ್ನ D ಅಂತ ಕರೆಯೋಣ. And the last one is B, that is growth of corpus luteum. So ಈ ಒಂದು ಆಪ್ಷನ್ ಇರೋದು ಯಾವುದ್ರಲ್ಲಿ ಅಂತ ಹೇಳಿದ್ರೆ in the option D. So ಈ ಎಲ್ಲ ಆಪ್ಷನ್ಸ್ ಇರೋದು D ನಲ್ಲಿ, therefore the right option is D. ಇದು ನೋಡಿ, this is the various events of menstrual cycle. Identify the correct sequence of hormones secreted during menstrual cycle in the human female. ಇಲ್ಲಿ ನೋಡಿ, ಮೆನ್ಸ್ಟ್ರೇಷನ್ ಫೇಸ್ ಆಗಬೇಕಾದ್ರೆ ಫಾಲಿಕಲ್ FSH ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತೆ. ಅದೇನು ಮಾಡುತ್ತೆ ಅಂದರೆ ಇಟ್ ಇಂಡ್ಯೂಸಸ್ ಫಾಲಿಕ್ಯುಲರ್ ಮೆಚುರೇಷನ್. ಈ ಫಾಲಿಕ್ಯುಲರ್ ಮೆಚುರೇಷನ್ ಆಗಬೇಕಾದ್ರೆ ಈ ಗ್ರ್ಯಾನ್ಯುಲರ್ ಸೆಲ್ಸ್ ಏನು ಮಾಡ್ತವೆ ಅಂದರೆ ಈಸ್ಟ್ರೋಜನ್ ಸೆಕ್ರೆಟ್ ಮಾಡ್ತವೆ, ಹೌದಾ. ಆಮೇಲೆ ನೆಕ್ಸ್ಟ್ ಇವೆಂಟ್ ಬರೋದೇ ಇನ್ಕ್ರೀಸ್ ಇನ್ ದ LH. ಆ LH ಏನು ಮಾಡುತ್ತೆ ಅಂದರೆ ಇಟ್ ಬ್ರಿಂಗ್ಸ್ ಅಬೌಟ್ ಓವ್ಯುಲೇಷನ್. ಆವಾಗ LH ಕಾನ್ಸಂಟ್ರೇಷನ್ ಜಾಸ್ತಿ ಆಗಿರುತ್ತೆ ಈ ಒಂದು ಸ್ಟೇಜಲ್ಲಿ. ಆಮೇಲೆ ಈ ಓವ್ಯುಲೇಷನ್ಸ್ ಆದಮೇಲೆ ಕಾರ್ಪಸ್ ಲ್ಯೂಟಿಯಂ ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೆ. ಈ ಕಾರ್ಪಸ್ ಲ್ಯೂಟಿಯಂ ಫಂಕ್ಷನ್ ಅಂತ ಹೇಳಿದ್ರೆ ಸೆಕ್ರೇಷನ್ ಆಫ್ ಪ್ರೊಜೆಸ್ಟ್ರಾನ್ ಫಾರ್ ದ ಮೇಂಟೆನೆನ್ಸ್ ಆಫ್ ಪ್ರೆಗ್ನೆನ್ಸಿ, ದೇರ್ಫೋರ್ ಕಾಲ್ಡ್ ಪ್ರೆಗ್ನೆನ್ಸಿ ಹಾರ್ಮೋನ್. ಹಾಗಾದ್ರೆ ಇದನ್ನ ನೋಡಿದ್ರೆ ಹಾರ್ಮೋನ್ಸ್ ಒಂದು ಆರ್ಡರ್ ಬರ್ತಕ್ಕಂಥ ರೀತಿ ಯಾವುದು ಅಂದರೆ ಫಸ್ಟ್ FSH ಬರುತ್ತೆ ಈ ಒಂದು ಇವೆಂಟ್ ಅಲ್ಲಿ, ನೆಕ್ಸ್ಟ್ ಈಸ್ಟ್ರೋಜನ್, ಹಾಗೆ LH ಬರಬೇಕು, ನೆಕ್ಸ್ಟ್ ಪ್ರೊಜೆಸ್ಟ್ರಾನ್ ಬರಬೇಕು. ಹಾಗಾದ್ರೆ ಎಲ್ಲ ಆಪ್ಷನ್ಸ್ ಇರ್ತಕ್ಕಂಥ ಒಂದು ಆಪ್ಷನ್ ಯಾವುದು ಅಂತ ಹೇಳಿದ್ರೆ that is B, FSH, estrogen, LH and progesterone. Therefore this is the right option. ಹಾಗೆ the decline in the levels of which hormone causes menstruation in females. ನಾನು ಹೇಳ್ತಾ ಇದ್ದೆ, ಸೊ ಮೆನ್ಸ್ಟ್ರೇಷನ್ ಆಗೋದಕ್ಕೆ ಮುಖ್ಯ ಕಾರಣ ಯಾವುದು ಅಂತ ಹೇಳಿದ್ರೆ LH as well as progesterone. ಯಾಕೆ ಅಂತ ಹೇಳಿದ್ರೆ LH maintains corpus luteum for the continuous secretion of progesterone. ಈ ಪ್ರೊಜೆಸ್ಟ್ರಾನ್ ಆಗಬೇಕಲ್ವಾ, ಅದಕ್ಕೋಸ್ಕರ ಏನು ಮಾಡುತ್ತೆ, ಈ LH ಪ್ರೊಜೆಸ್ಟ್ರಾನ್ ಸೆಕ್ರೆಟ್ ಮಾಡೋದಕ್ಕೆ ಕಾರ್ಪಸ್ ಲ್ಯೂಟಿಯಂನ ಮೇಂಟೈನ್ ಮಾಡುತ್ತೆ. ಈ ಪ್ರೊಜೆಸ್ಟ್ರಾನ್ ಏನು ಮಾಡುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ ಇಟ್ ಮೇಂಟೈನ್ಸ್ ಪ್ರೆಗ್ನೆನ್ಸಿ. ಈಗ ಅಲ್ಲಿ ಪ್ರೆಗ್ನೆನ್ಸಿನೇ ಆಗಿಲ್ಲ, ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗಿಲ್ಲ ಅಂತ ಹೇಳಿದ್ರೆ ದ ಲೆವೆಲ್ಸ್ ಆಫ್ LH ಆಟೋಮೆಟಿಕ್ಲಿ ಡಿಕ್ಲೈನ್ಸ್. LH ಡಿಕ್ಲೈನ್ಸ್ ಆದಮೇಲೆ ಈ ಕಾರ್ಪಸ್ ಲ್ಯೂಟಿಯಂನ ಮೇಂಟೈನ್ ಮಾಡೋದಕ್ಕಾಗಲ್ಲ. ಅವಾಗ ಏನಾಗುತ್ತೆ, ಕಾರ್ಪಸ್ ಲ್ಯೂಟಿಯಂ ಅಂಡರ್ಗೋ ಡಿಜೆನರೇಷನ್. ಕಾರ್ಪಸ್ ಲ್ಯೂಟಿಯಂ ಡಿಜೆನರೇಷನ್ ಆದರೆ ಪ್ರೊಜೆಸ್ಟ್ರಾನ್ ಲೆವೆಲ್ ಏನಾಗುತ್ತೆ, ಕಡಿಮೆ ಆಗುತ್ತೆ. ಸಡನ್ ಡಿಕ್ಲೈನ್ ದ ಲೆವೆಲ್ಸ್ ಆಫ್ ಪ್ರೊಜೆಸ್ಟ್ರಾನ್ ಈಸ್ ಇಮಿಡಿಯೇಟ್ ಕಾಸ್ ಆಫ್ ಮೆನ್ಸ್ಟ್ರೇಷನ್. ಬಟ್ ಇಲ್ಲಿ ಪ್ರೊಜೆಸ್ಟ್ರಾನ್ ಕೊಟ್ಟಿಲ್ಲ, ಇಲ್ಲಿ ಕೊಡ್ತಿರ್ತಕ್ಕಂಥ ಆಪ್ಷನ್ ಯಾವುದು ಅಂತ ಹೇಳಿದ್ರೆ LH. ಸೊ therefore this is the right option, LH. Gonadotrophins refers to ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ gonadotropins ಆರ್ ದೋಸ್ ದೇ ಆಕ್ಟ್ ಆನ್ ಗೋನಾಡ್ಸ್. ಅದು ಟೆಸ್ಟಿಸ್ ಮತ್ತು ಓರಿನ ಗೊನಾಡ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಅದ್ರ ಮೇಲೆ ಆಕ್ಟ್ ಮಾಡ್ತಕ್ಕಂಥ ಒಂದು ಹಾರ್ಮೋನ್ಸ್ಗೆ ನಾವು gonadotropins ಅಂತ ಕರಿತೀವಿ. ದಿಸ್ ಅ ಡೈರೆಕ್ಟ್ ಕ್ವಶನ್. ಅಲ್ಲಿ LH ಮತ್ತು FSH. ಸೊ ದೇರ್ಫೋರ್ ದ ರೈಟ್ ಆಪ್ಷನ್ ಈಸ್ ಎ, ಲ್ಯೂಟಿನೈಸಿಂಗ್ ಹಾರ್ಮೋನ್ ಅಂಡ್ follicle stimulating hormone. The number of follower bodies formed at the first meiotic division during oogenesis. oogenesis ಆಗಬೇಕಾದ್ರೆ polar bodies ಎಷ್ಟು ಬರುತ್ತೆ ಯಾವಾಗ ಅಂತ ಹೇಳಿದ್ರೆ ಅಟ್ಸ್ ದ ಫಸ್ಟ್ ಮಿಯಾಟಿಕ್ ಡಿವಿಷನ್. ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ ಪ್ರೈಮರಿ oocyte ಅಂಡರ್ಗೋ ಫಸ್ಟ್ ಮಿಯಾಸಿಸ್, ಇಟ್ಸ್ ಅನ್ಈಕ್ವಲ್. ಆಸ್ ಎ ರಿಸಲ್ಟ್ ಒಂದು ಸೆಕೆಂಡರಿ oocyte ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೆ, ಒಂದು ಪೋಲಾರ್ ಬಾಡಿ ಬರುತ್ತೆ. ಇದನ್ನ ಫಸ್ಟ್ ಪೋಲಾರ್ ಬಾಡಿ ಅಂತ ಕರಿತೀವಿ, ಇಲ್ಲ ಫಸ್ಟ್ ಪೋಲೋಸೈಟ್ ಅಂತ ಕರಿತೀವಿ. ಹಾಗಾದ್ರೆ ಈ ಆಪ್ಷನ್ಸ್ ಅಲ್ಲಿ ಒಂದು ಇಲ್ಲಿದೆ ನೋಡಿ. ಸೊ therefore the number of polar bodies formed at the end of first meiotic division during oogenesis is only 1. So therefore A is the right option. When does the secondary oocyte completes its second meiotic division? ಹಾಗಾದ್ರೆ ಓವ್ಯುಲೇಷನ್ ಆಗಬೇಕಾದ್ರೆ ಎಗ್ ಯಾವ ಸ್ಟೇಜಲ್ಲಿ ಇರುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ ಸೆಕೆಂಡರಿ oocyte ಸ್ಟೇಜಲ್ಲಿ ಇರುತ್ತೆ. ಅದಿನ್ನೂ ಫಸ್ಟ್ ಮಿಯಾಸಿಸ್ ಕಂಪ್ಲೀಟ್ ಆಗಿರುತ್ತೆ ಅಥವಾ ಮೆಟಾಫೇಸ್ 1 ಸ್ಟೇಜಲ್ಲಿ ಇರುತ್ತೆ. ಅದು ಮಿಯಾಸಿಸ್ 1 ಅಲ್ಲೇ ಇರೋದ್ರಿಂದ ಅದು ಮಿಯಾಸಿಸ್ 2ನ ಕಂಪ್ಲೀಟ್ ಮಾಡ್ಕೋಬೇಕು. ಮಿಯಾಸಿಸ್ 2 ಕಂಪ್ಲೀಟ್ ಆಗೋದು ಯಾವಾಗ ಅಂತ ಹೇಳಿದ್ರೆ ವೆನ್ ಸ್ಪರ್ಮ್ ಗೇನ್ಸ್ ಎಂಟ್ರಿ ಇನ್ ಟು ದ ಎಗ್. ಈಗ ಹೆಗ್ಗಿದ್ದಾಗ ಸ್ಪರ್ಮ್ ಎಂಟ್ರಿ ಆಗುತ್ತೆ. ಎಂಟ್ರಿ ಆದಾಗ ಈ ಸೆಕೆಂಡರಿ oocyte ಅಂತ ಏನು ಕರಿತೀವಿ, ಇಟ್ ಕಂಪ್ಲೀಟ್ಸ್ ಇಟ್ ಸೆಕೆಂಡ್ ಮಿಯಾಸಿಸ್. ಸೆಕೆಂಡ್ ಮಿಯಾಸಿಸ್ ಆಗಬೇಕಾದ್ರೆ ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ oogenesis ಅಲ್ಲಿ ಪೋಲಾರ್ ಬಾಡೀಸ್ ಎಲ್ಲ ರಿಲೀಸ್ ಆಗುತ್ತೆ. ಈ ರಿಲೀಸ್ ಆದಾಗ ಈ ಪೋಲಾರ್ ಬಾಡೀಸ್ ಎಂಟರ್ಸ್ ಇಂಟು ಪೆರಿವಿಟಲೈನ್ ಸ್ಪೇಸ್. ಹಾಗೆ ಈ ಸೆಕೆಂಡರಿ oocyte ಅಂತ ಏನು ಕರಿತೀವಿ, ಇಟ್ ಕಂಪ್ಲೀಟ್ಸ್ ಇಟ್ ಸೆಕೆಂಡ್ ಮಿಯಾಸಿಸ್, ದೇರ್ ಬೈ ಇಟ್ ಬಿಕಮ್ಸ್ ooted. ಸೊ ಅದಕ್ಕೆ ಇಲ್ಲಿ ಅದನ್ನ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತಕ್ಕಂಥ ಆಪ್ಷನ್ ಯಾವ್ದು ಅಂತ ಹೇಳಿದ್ರೆ ಆಫ್ಟರ್ ದ ಎಂಟ್ರಿ ಆಫ್ ಸ್ಪರ್ಮ್ before fertilization. So therefore the right option is A, after the entry of sperm and before fertilization. What is the number of egg cells and polar bodies formed from a primary oocyte during oogenesis? You know that during oogenesis primary oocyte undergo first meiosis and second meiosis. As a result, so it produces 1 egg are ooted and 3 polar bodies. ಇದು ಯಾವ ಕಾನ್ಸೆಪ್ಟ್ ಮೇಲೆ ಬೇಸ್ ಆಗಿದೆ ಅಂದ್ರೆ oogenesis. ಈ ಒಂದು ಸ್ಕೀಮ್ಯಾಟಿಕ್ ರೆಪ್ರೆಸೆಂಟೇಷನ್ ನಿಮ್ಮ ಮೆಂಟಲ್ ಪಿಕ್ಚರ್ ಅಲ್ಲಿ ಇದ್ರೆ ಈಸಿಯಾಗಿ ಆನ್ಸರ್ ಮಾಡಬಹುದು. ಅಂದ್ರೆ oogenesis ಆದ್ರೆ ಎಷ್ಟು ಎಗ್ ಅಥವಾ ಓಮ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗುತ್ತೆ ಮತ್ತು ಎಷ್ಟು ಪೋಲಾರ್ ಬಾಡಿ ಆಗುತ್ತೆ ಅಂತ ಹೇಳಿಬಿಟ್ಟು. ಸೊ 1 egg and 3 polar bodies. So therefore A is the right option. When does extrusion of second polar body from egg nucleus occurs? ನಾನು ಪ್ರೀವಿಯಸ್ ಕ್ವಶನ್ ಅಲ್ಲಿ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತಾ ಇದ್ದೆ. When sperm enters into the cytoplasm of the egg, the first event is completion of second meiosis by secondary oocyte. ಆ ಸೆಕೆಂಡರಿ oocyte ಕಂಪ್ಲೀಟ್ ಆಗಬೇಕು, ಸೆಕೆಂಡ್ ಮಿಯಾಸಿಸ್ ಆಗಬೇಕಾದ್ರೆ ಪೋಲಾರ್ ಬಾಡಿ ಕೂಡ ರಿಲೀಸ್ ಆಗುತ್ತೆ. ಆ ಪೋಲಾರ್ ಬಾಡೀಸ್ ಆ ಒಂದು ಸ್ಟೇಜ್ ಅಲ್ಲಿ ಹೊರಗಡೆ ಹೋಗಬೇಕು, ಅದಕ್ಕೆ ನಾವು ಎಕ್ಸ್ಟ್ರೂಷನ್ ಅಂತ ಕರಿತೀವಿ. ಸೊ therefore the correct option is A, after the entry of sperm before completion of fertilization. ಮಿಕ್ಕಿರ್ತಕ್ಕಂಥ ಆಪ್ಷನ್ಸ್ ದೇ ಆರ್ ಅಷ್ಟು ಕರೆಕ್ಟ್ ಆಗಿರೋದಿಲ್ಲ, ಅಂದ್ರೆ ಕರೆಕ್ಟ್ ಆಗಿರೋದಿಲ್ಲ. ಸೊ ದ ರೈಟ್ ಆಪ್ಷನ್ ಈಸ್ A. Choose the correct sequence of human embryonic development. ಹ್ಯೂಮನ್ ಎಂಬ್ರಿಯೋ ಡೆವಲಪ್ ಆಗಬೇಕಾದ್ರೆ ಏನೇನು ಈವೆಂಟ್ಸ್ ಬರುತ್ತೆ ಒಂದು ಆರ್ಡರ್ಲಿ. ಇಲ್ಲಿ ಫಸ್ಟ್ ಹಾಗೆ ನಮ್ಗೆ ಗೊತ್ತಿದೆ, ನಮಗೆ ಈ ಒಂದು ಕ್ವಶನ್ ಆನ್ಸರ್ ಮಾಡಬೇಕಾದ್ರೆ ನಮಗೆ ವಿ ಶುಡ್ ನೋ ಐಡಿಯಾ ಅಬೌಟ್ ಪ್ರೀ ಫರ್ಟಿಲೈಸೇಷನ್ ಅಂಡ್ ಪೋಸ್ಟ್ ಫರ್ಟಿಲೈಸೇಷನ್ ಈವೆಂಟ್ಸ್. Insemination. Insemination is a process of release of sperms into the vagina during sexual intercourse. So that leads to fertilization. ಅದೇ ಟೈಮಲ್ಲಿ ಸ್ಪರ್ಮ್ ಮೀಟ್ ಮಾಡಿದ್ರೆ ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗುತ್ತೆ. ಫರ್ಟಿಲೈಸೇಷನ್ ಲೀಡ್ಸ್ ಟು zygote. Zygote undergo cleavage. That is a repeated mitotic division. It results in the formation of morula, blastula and so on. Then there is the implantation of embryo. Then organogenesis, the process of formation of organs. The complete duration required for the development of embryo is gestation period. After gestation period, childbirth. So ಈ ಒಂದು ಆಪ್ಷನ್ಸ್ ಅಲ್ಲಿ ನಾವೆಲ್ಲನೂ ನೋಡ್ತೀವಿ, ಯಾಕಂದ್ರೆ ಇನ್ಸೆಮಿನೇಷನ್ ಬಿಗಿನಿಂಗ್ ಇರೋದ್ರಿಂದ we can predict. So therefore the right option is A. The hormones which acts on Sertoli cells and stimulate the process of spermiogenesis is. ಇದು gonadotropin ಹಾರ್ಮೋನ್ಸ್ ಮೇಲೆ ಬೇಸ್ ಆಗಿದೆ. ಇಲ್ಲಿ the hormone which acts on Sertoli cells. Sertoli ನನ್ನ ಹಾರ್ಮೋನ್ ಕಂಟ್ರೋಲ್ ಆಫ್ ಸ್ಪರ್ಮಟೋಜೆನಿಸಿಸ್ ಹೇಳ್ಬೇಕಾದ್ರೆ ಹೇಳ್ತಾ ಇದೆ. Follicle stimulating hormone acts on Sertoli cells for the secretion of certain factors. So those factors are essential for transformation of spermatids into sperm. This process is called spermiogenesis. But LH ಏನಾಗುತ್ತೆ ಅಂದರೆ it acts on Leydig cells for the secretion of testosterone, which in turn stimulates spermatogenesis. ಅದಕ್ಕೋಸ್ಕರ the right option is FSH, follicle stimulating hormone. Human placenta is formed by, ನಾನು ಹೇಳ್ತಾ ಇದ್ದೆ, human placenta is formed by tissues of the fetus as well as mother. The mother part of the placenta is uterine epithelium. ಇಲ್ಲಿ ನೋಡ್ತೀವಿ, ಇಲ್ಲಿ ಒಂದು ಆಪ್ಷನ್ಸ್ ಅಲ್ಲಿ ಇದೆ.
and fetal part of the placenta is chorion as well as allantois. Chorion barutte matte allantois kuda barutte. Chorion form finger like projections called chorion equili, and the blood vessels produced by allantois extended into it. ಅದಕ್ಕೋಸ್ಕರ ಕೊರಿಯಾನು ಬೇಕು, ಅಲಂಟಾಯ್ಸು ಬೇಕು, ಮತ್ತು ಯುಟ್ರೈನ್ ವಾಲ್ ಕೂಡ ಬೇಕಾಗುತ್ತೆ. ಇಟ್ಸ್ maternal part of the placenta. So therefore the right option is D, chorion, allantois and uterine wall. Human placenta is, ನಾನು ಹೇಳ್ತಾ ಇದ್ದೆ ನೇಚರ್ ಆಫ್ ಹ್ಯೂಮನ್ ಪ್ಲಾಸೆಂಟಾ ಅಂತ ಹೇಳಿದ್ರೆ, ಹ್ಯೂಮನ್ ಪ್ಲಾಸೆಂಟಾ ಇಸ್ ಹ್ಯೂಮಕೋರಿಯಲ್. ಯಾಕಂತ ಹೇಳಿದ್ರೆ chorion equili are dipped in the uterine epithelium, and that means they are bathed in maternal blood. So that's why it is said to be hemocorial, since placenta formation requires both chorion as well as allantois. Both chorion and allantois take part in the process of formation of placenta. So therefore it is said to be choreo-allantoic placenta anta karitivi. But at the time of parturition, placenta automatically separates, but umbilical cord is still attached to the fetus. It is later separated by gynecologist or birth attendants. So therefore all options ಬೇಕಾಗುತ್ತೆ. The right option is D. Identify the hormones A, B, C that are labeled in the given diagram. ಇಲ್ಲಿ ನಾವು ಗೊತ್ತಿದೆ, ಇಟ್ ಈಸ್ ಎ ಕ್ವಶನ್ ಆನ್ ಹಾರ್ಮೋನ್ ರೆಗ್ಯುಲೇಷನ್ ಆಫ್ ಸ್ಪರ್ಮೆಟೋಜೆನಿಸಿಸ್. ಇಲ್ಲಿ ನೋಡಿ, ಹೈಪೋಥಾಲಮಸ್ ಸೆಕ್ರೀಟ್ಸ್ ಹಾರ್ಮೋನ್ಸ್. ಆ ಹಾರ್ಮೋನ್ಸು ಗೋನಾಡೋಟ್ರೋಫಿನ್ ರಿಲೀಸಿಂಗ್ ಹಾರ್ಮೋನ್. ಇಟ್ ಆಕ್ಟ್ಸ್ ಆನ್ ಪಿಟ್ಯೂಟರಿ ಫಾರ್ ದ ಸೆಕ್ರೀಷನ್ ಆಫ್ 2 ಗೋನಾಡೋಟ್ರೋಫಿನ್ಸ್, ನೇಮ್ಲಿ LH. LH acts on Leydig cells, and FSH it acts on Sertoli cells. ಈ ಎರಡು ಈ ಆಪ್ಷನ್ಸ್ ಇರ್ತಕ್ಕಂಥದ್ದು ಯಾವುದ್ರಲ್ಲಿ ಅಂದರೆ ಬಿನಲ್ಲಿ LH ಇದೆ ಮತ್ತು FSH ಕೂಡ ಇದೆ. ಸೊ therefore the right option is B.
Which of the following extraembryonic membranes forms the major part of the placenta in mammals? ಇಲ್ಲಿ major part of the placenta is from fetal side. ಅದನ್ನ ನಾವು ಕಾಮನ್ ಆಗಿ chorion ಅಂತ ಕರಿತೀವಿ. You know that it forms chorion equili. As a result, fetus is attached to the wall of the endometrium with help of chorion equili or through placenta. Therefore, the right option is chorion. Amnion is the membrane immediately surrounding the fetus. Allantois is another fetal membrane. Other one is yolk sac. The right option is A. ನೋಡಿ, ಇದ್ರ ಬಗ್ಗೆ ಸ್ವಲ್ಪ ಎಕ್ಸ್ಟ್ರಾ ಎಂಬ್ರಿಯಾನಿಕ್ ಮೆಂಬ್ರೇನ್ಸ್ ಬಗ್ಗೆ ಸ್ವಲ್ಪ ಐಡಿಯಾ ಬೇಕಾಗುತ್ತೆ. ಎಕ್ಸ್ಟ್ರಾ ಎಂಬ್ರಿಯಾನಿಕ್ ಮೆಂಬ್ರೇನ್ಸ್ ಯಾವುದು ಅಂತ ಹೇಳಿದ್ರೆ, ನೋಡಿ ಈ ಒಂದು ಡಯಾಗ್ರಾಮೆಟಿಕ್ ರೆಪ್ರೆಸೆಂಟೇಷನ್ ನೋಡ್ಬೋದು. ದಿಸ್ ದ ಎಂಬ್ರಿಯೋ. ಎಂಬ್ರಿಯೋ ಇಸ್ ಇಮಿಡಿಯೇಟ್ಲಿ ಎಂಬ್ರಿಯೋ. ಔಟರ್ ಮೋಸ್ಟ್ ಲೇಯರ್ಗೆ ನಾವು ಕೋರಿಯನ್ ಅಂತ ಕರಿತೀವಿ. ಇಟ್ ಇಸ್ ಔಟರ್ ಮೋಸ್ಟ್ ಫೀಟಲ್ ಮೆಂಬ್ರೇನ್ ಅಂಡ್ ಇಟ್ ಫಾರ್ಮ್ ಪ್ಲಾಸೆಂಟಾ. ಅದೇ ರೀತಿ ನೆಕ್ಸ್ಟ್ ಆಮ್ನಿಯನ್ ಬರುತ್ತೆ. ಇಮಿಡಿಯೇಟ್ಲಿ ಅದು ಎಂಬ್ರಿಯೋ ಸುತ್ತ ಇರ್ತಕ್ಕಂಥ ಮೆಂಬ್ರೇನ್. ಇಟ್ ಇಸ್ ಇನ್ನರ್ ಮೋಸ್ಟ್ ಫೀಟಲ್ ಮೆಂಬ್ರೇನ್ ಅಂಡ್ immediately surrounds the embryo. The space present between the embryo and amnion is called amniotic cavity, filled with amniotic fluid that protects the embryo from external shocks and mechanical injury. ಹಾಗೆ the space ನಾವು amniotic cavity ಅಂತ ಕರಿತೀವಿ. The next one is called yolk sac. It is non-functional or remnant in mammals. Because it's not necessary yolk synthesis. It produces corpuscles or blood cells till the liver becomes functional. And other extraembryonic membrane is allantois, develops in the area of gut and produces blood vessels. ಅದಕ್ಕೆ ನಾವು ಕೋರಿಯಾನೋ ಫಾರ್ಮೇಷನ್ ಬೇಕು ಪ್ಲಾಸೆಂಟಾ ಫಾರ್ಮೇಷನ್ಸ್ ಅಲ್ಲಿ, ಅಲಂಟಾಯ್ಸು ಕೂಡ ಬೇಕಾಗುತ್ತೆ. ಅದಕ್ಕೋಸ್ಕರ ಹ್ಯೂಮನ್ ಪ್ಲಾಸೆಂಟಾ ಇಸ್ ಕಾಲ್ಡ್ ಕೋರಿಯೋ ಅಲಂಟಾಯಿಕ್ ಪ್ಲಾಸೆಂಟಾ. Look at the next question. Which of the following extraembryonic membranes forms the major placenta in mammals? ಇಲ್ಲಿ major part is chorion. Just now I have explained. So this is the right option. The extraembryonic membranes of the mammalian embryo are derived from. ನಾನು ಬ್ಲಾಸ್ಟೋಸಿಸ್ಟ್ ಹೇಳ್ಬೇಕಾದ್ರೆ ಹೇಳ್ತಾ ಇದ್ದೆ, ಆ ಬ್ಲಾಸ್ಟೋಸಿಸ್ಟ್ ಅಲ್ಲಿ 2 ಲೇಯರ್ಸ್ ಬರುತ್ತೆ, ಟ್ರೋಪೋಎಕ್ಟೋಡರ್ಮ್ ಅಂಡ್ ಇನ್ನರ್ ಸೆಲ್ ಮಾಸ್ ಅಂತ ಕರಿತೀವಿ. ಆ ಟ್ರೋಪೋಎಕ್ಟೋಡರ್ಮ್ ರಿಸಲ್ಟ್ಸ್ ಇನ್ ದ ಫಾರ್ಮೇಷನ್ ಆಫ್ ಪ್ಲಾಸೆಂಟಾ as well as extraembryonic membranes. So therefore trophoblast is the right options and the rest are wrong. ಯಾಕಂದ್ರೆ embryoblast develops into embryo, Leydig cells secretes testosterone, and Sertoli cells are nutritive in function. Which of the following hormone is not produced in women during pregnancy? ಇದು ಒಂದು HCG ಇದೆ, human chorionic gonadotrophin, human placental lactogen, relaxin and inhibin. Placenta acts as a temporary endocrine gland, secreting some of the hormones like HCG, HPL and relaxin. But inhibin is secreted by Sertoli cells, so therefore the right option is D. It is not secreted by placenta. Match the hormones given in column 1 with their functions given in column 2. Choose the answer from the options given below. ಇಲ್ಲಿ ಒಂದು ಸೈಡ್ ಹಾರ್ಮೋನ್ಸ್ ಕೊಟ್ಟಿದ್ದಾರೆ, ಅದರ್ ಸೈಡ್ ಫಂಕ್ಷನ್ಸ್ ಕೊಟ್ಟಿದ್ದಾರೆ. ನೀವು ಈಸಿಯಾಗಿ ಪ್ರಿಡಿಕ್ಟ್ ಮಾಡ್ಬೋದು. is the hormone induces ovulation in females. ಹಾಗಾದರೆ LH ಗೆ ಆಪ್ಷನ್ ಯಾವುದು? ಎಸ್ ಬರುತ್ತೆ ಇಲ್ಲಿ. ನೆಕ್ಸ್ಟು FSH, ಇಟ್ ಇಸ್ ಮೆಂಟ್ ಫಾರ್ ಫಾಲಿಕ್ಯುಲರ್ ಮೆಚುರೇಷನ್. ಹಾಗಾದರೆ ಎಫ್ ಗೆ ಆಪ್ಷನ್ ಯಾವುದು ಅಂತ ಹೇಳಿದ್ರೆ ಆರ್ ಅಂತ ಕರಿತೀವಿ. ಪ್ರೊಜೆಸ್ಟ್ರಾನ್, ಇಟ್ಸ್ ಎ ಪ್ರೆಗ್ನೆನ್ಸಿ ಹಾರ್ಮೋನ್. ಸೊ ದೇರ್ಫೋರ್ ಇದಕ್ಕೆ ಕ್ಯೂ ಬರಬೇಕಾಗುತ್ತೆ. ನೆಕ್ಸ್ಟ್ ಆಕ್ಸಿಟೋಸಿನ್, ಇಟ್ಸ್ ಎ ಬರ್ತ್ ಹಾರ್ಮೋನ್ ಸಿನ್ಸ್ ಇಟ್ ಹೆಲ್ಪ್ಸ್ ಇನ್ ಪಾರ್ಚುರೇಷನ್. ಅದಕ್ಕೆ ಪಿ ಆಪ್ಷನ್ ಬರುತ್ತೆ. ಪ್ರೊಲ್ಯಾಕ್ಟಿನ್, ಇಟ್ಸ್ ಎ ಮಿಲ್ಕ್ ಸಿಂಥಸೈಸಿಂಗ್ ಆರ್ ಸೆಕ್ರೇಟಿಂಗ್ ಹಾರ್ಮೋನ್. ಅದು ಕಾಮನ್ ಆಗಿ ಮಿಲ್ಕ್ ಪ್ರೊಡಕ್ಷನ್, ದಟ್ ಇಸ್ ಟಿ. ಆಲ್ ದ ಆಪ್ಷನ್ಸ್ ಯಾವುದರಲ್ಲಿ ಇದೆ ನೋಡಿ. So it is in A, so therefore this is a right option. Yeah, let us take up next chapter, reproductive health. Reproductive health refers to healthy reproductive organs with normal functions. According to World Health Organization, reproductive health is a total well-being in all aspects of reproduction such as physical, emotional, behavioral and social. Reproductively healthy society. A society with people having physically and functionally normal reproductive organs and normal emotional and behavioral interactions among them in all sex-related aspects. Idu now reproductively healthy society anta kariತೀವಿ. In fact, India is the first country in the world to consider reproductive health as its goal. As a result, it initiated several programs to achieve it. And one such program is family planning program. It was initiated in the year 1951. It's an understanding between the couples to limit the size of a family or to space or postpone pregnancy. Such several programs were initiated to achieve reproductive health of the society, and all such successful programs were in operations under a particular name called RCH, Reproductive and Child Healthcare Program. The major goals of RCH programs are creating awareness among people about sexually related aspects such as proper information about reproductive organs, adolescence and related changes, safe and hygienic sexual practices, sexually transmitted diseases, about available birth control options, care of pregnant mothers, postnatal care of pregnant mother and child, importance of breastfeeding and vaccination. Other goal is providing facilities and support for building up reproductively healthy and socially responsible society. Assistance and care to people in reproduction related problems like pregnancy, delivery, STDs, abortions, contraception, menstrual problems and infertility. Implementation of better techniques and new strategies from time to time, and one such example is Saheli. It's a new oral contraceptive developed by CDRI Lucknow, Central Drug Research Lucknow. The other technique is amniocentesis. It's a feet sex determination test based on the chromosomes present in the amniotic cells surrounding the fetus. Indicators of improved reproductive health of the society. Better awareness about sex-related matters, increased number of medically assisted deliveries, better postnatal care leading to increased maternal and infant mortality rate, increased number of couples with small family norms, better detection and cure of sexually transmitted diseases, overall increase better medical and health care facilities for all sex-related problems. ಇವೇನು ತೋರಿಸುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ ಒಂದು ರಿಪ್ರೊಡಕ್ಟಿವ್ ಸೊಸೈಟಿನಲ್ಲಿ ಒಂದು ರಿಪ್ರೊಡಕ್ಟಿವ್ ಹೆಲ್ತ್ ಅಂತ ಏನು ಕರಿತೀವಿ ಅದು ಇಂಪ್ರೂವ್ ಆಗಿದೆ ಅಂತ ತೋರಿಸ್ತಾರೆ. ಸೊ ದೀಸ್ ಆರ್ ಇಂಡಿಸಿಸ್ ಆರ್ ಇಂಡಿಕೇಟರ್ಸ್. ಪಾಪ್ಯುಲೇಷನ್ ಎಕ್ಸ್ಪ್ಲೋಷನ್ ಅಂಡ್ ಬರ್ತ್ ಕಂಟ್ರೋಲ್, ಬೆಟರ್ ಮೆಡಿಕಲ್, ದೀಸ್ ಆರ್ ದ ಕಾಸಸ್. ಪಾಪ್ಯುಲೇಷನ್ ಎಕ್ಸ್ಪ್ಲೋಷನ್ ಆಗೋದಕ್ಕೆ ಕಾರಣಗಳು ಬೆಟರ್ ಮೆಡಿಕಲ್ ಅಂಡ್ ಹೆಲ್ತ್ ಕೇರ್ ಫೆಸಿಲಿಟೀಸ್, ರ್ಯಾಪಿಡ್ ಡಿಕ್ಲೈನ್ ಇನ್ ಮಾರ್ಟಾಲಿಟಿ ಆರ್ ಡೆತ್ ರೇಟ್, decrease in maternal mortality rate, decrease in infant mortality rate, increase in the number of people with reproducible age, lack of epidemics, better detection and cure available for almost all the diseases due to improved and medical healthcare facilities. Measures to check or tackle rapid population growth, motivate smaller families by using various contraceptive methods, incentives given to couples with small family norms, statutory rising of marriageable age of the male to 21 years and that of females to 18 years. And the best method to prevent overpopulation is to adapt contraceptive methods. What is contraceptive method? The birth control methods which prevents unwanted fertilization and pregnancy are said to be contraceptive methods. So contraceptive methods are of two types, namely temporary methods and permanent methods. In temporary methods, the fertility comes to normal after the disuse of these contraceptives. So these includes natural methods, then physical barriers, intrauterine devices,
contraceptive pills, injections as well as implants. ಇವೆಲ್ಲ ಕೂಡ ನಾವು temporary methods ಅಂತ ಕರಿತೀವಿ. And permanent methods are surgical methods. They may lead to complete sterility as reversibility is very poor with them. And two such permanent methods are sterilization procedures, namely vasectomy and tubectomy. Vasectomy in males and tubectomy in females. Natural methods. The natural methods of contraception includes periodic abstinence, withdrawal or coitus interrupts, and lactational amenorrhea. How do these natural methods act as contraceptives? Natural methods work on the principle of avoiding sperm and egg meeting. That is how they act as a contraceptives. Then barrier methods includes condoms in males, diaphragm or cervical caps in females, vaults in females. How do barrier methods prevent fertilization? Barrier methods act as a contraceptives and thereby they prevent fertilization and pregnancy by preventing physical meeting of sperm and egg during sexual intercourse. One of the advantages of barrier methods like condoms is very important, and rather than the ಏನು ಪ್ರಯೋಜನ ಅಂತ ಹೇಳಿದ್ರೆ, so these barrier methods also prevent the transmission of sexually transmitted diseases during sexual intercourse. Intrauterine devices. Intrauterine devices are the plastic or metallic objects which are inserted into uterus by expert gynecologists. So these devices remain in the same place for many years till female desires to postpone pregnancy. There are three different types of intrauterine devices, namely non-medicated IUDs, copper releasing IUDs, and hormone releasing IUDs. ಇಲ್ಲಿ non-medicated IUDs ಅಂದರೆ lips, loops ಮತ್ತು spirals. Copper releasing IUDs constitute copper T, copper 7 and multiload 375. Hormone releasing IUDs ಯಾವುದು ಅಂತ ಹೇಳಿದ್ರೆ progestasat and LNG 20. Then how do these intrauterine devices prevent fertilization or pregnancy? IUDs increase phagocytosis of sperm within the uterus. Copper releasing IUDs suppress sperm motility and fertilizing capacity of sperms. The hormone releasing IUDs prevent implantation of embryo and make cervix hostile to sperm. This is how they prevent fertilization or pregnancy. Oral pills. The contraceptive pills that are taken orally in the form of tablets are called oral pills. So they contain synthetic hormones like estrogens and progesterones. There are two types of pills, oral pills, namely mini pills and combined pills. Mini pill contains only progesterone, whereas combined pills contains both estrogen and progesterone. So these oral pills have to be taken orally continuously for about 21 days, preferably starting from 5th day. After a gap of 7 days, they should be repeated in the same manner till female desires to postpone pregnancy. Combined pill examples ಯಾವುದು ಅಂತ ಹೇಳಿದ್ರೆ ಇಲ್ಲಿ ಕೊಟ್ಟಿದ್ದಾರೆ ಮಾಲಾ ಡಿ ಅಂತ ಹೇಳ್ಬಿಟ್ಟು ಮತ್ತೆ ಮಾಲಾ ಎನ್. ಸೊ ದೀಸ್ ಆರ್ ದಿ ಎಕ್ಸಾಂಪಲ್ಸ್ ಫಾರ್ combined pills. Oral pills prevent ovulation and implantation of embryo, so and also alter the quality of cervical mucus to prevent or retard the entry of sperm. This is how they prevent fertilization and pregnancy. ಅಂದರೆ ಓವ್ಯುಲೇಷನ್ ಮತ್ತು ಇಂಪ್ಲಾಂಟೇಷನ್ ಆಗೋದಕ್ಕೆ ಬಿಡೋದಿಲ್ಲ. ಸೊ ದಟ್ಸ್ ವೈ ದೇ ಆರ್ ಸೆಡ್ ಟು ಬಿ ಕಾಂಟ್ರಾಸೆಪ್ಟಿವ್ ಮೆಥಡ್ಸ್. ಸಮಥಿಂಗ್ ಅಬೌಟ್ ಸಮ್ ನ್ಯೂ ಆಸ್ಪೆಕ್ಟ್ಸ್ ಆಫ್ ಸಹೇಲಿ. ಸ್ವಲ್ಪ ಸಹೇಲಿ ಬಗ್ಗೆ ನಾವು ತಿಳ್ಕೊಳ್ಳೋಣ. ಸಹೇಲಿ is a new oral contraceptive for females developed by CDRI Lucknow, Central Drug Research Lucknow. It contains non-steroidal preparation. ಇದಕ್ಕೆ ನಾವು ಸೆಂಡ್ಕ್ರೋಮ್ಯಾನ್ ಅಂತ ಕರಿತೀವಿ, which is taken once a week after an initial intake of twice a week dose for 3 months. ಅದಕ್ಕೋಸ್ಕರ ಇದನ್ನು ನಾವು once a week oral pill ಅಂತ ಕರಿತೀವಿ. Emergency contraceptive pills. Oral pills also can be used to overcome high risk of pregnancy condition. The oral pills which are used to overcome high risk pregnancy conditions due to rape, casual unprotected sex, or failure of the contraceptive used during coitus or sexual intercourse are said to be emergency contraceptive pills. Emergency contraceptive pills are also said to be morning after pills. The morning after pill is effective if taken within 72 hours of coitus up to 5 days after having unprotected sex. Injections and implants. Injections and implants also used by females as contraceptive devices. One such example is implants. Hormonal implants like Narplan are surgically implanted under the skin
for about 5 years to prevent fertilization and pregnancy. So therefore, they also act as a contraceptive device or methods. Surgical methods includes vasectomy in males and tubectomy in females. Vasectomy is a surgical procedure in which vasa deferens of both sides cut and tied to prevent the transport of sperm to the site of fertilization. In tubectomy, fallopian tubes of both sides is cut and tied to prevent the transport of egg or ovum to the site of fertilization. So, surgical intervention prevents the transport of both sperm as well as egg to the site of fertilization. So, that is how surgical methods or sterilization methods prevent fertilization. MTP, medical termination of pregnancy, it's also called induced abortion. Termination of pregnancy before full term maturation of the embryo is called MTP, that is medical termination of pregnancy. MTP is necessary to get rid of unwanted pregnancies, either due to casual unprotected intercourse or failure of the contraceptive used during sexual intercourse or coitus. Advised, MTP is advised in cases where continuation of pregnancy could be harmful or fatal, either to mother or to the fetus or both. If done, it is safe within 12 weeks of pregnancy, but it is legally banned in the country in the year 1971 to prevent its sterilization and indiscriminate killing of female feticides. Sexually transmitted diseases, abbreviated as STDs. So these are the diseases which spreads through sexual contact with the infected person. These are also called venereal diseases or VD, or reproductive tract infections or RTI. The common sexually transmitted diseases include gonorrhea, syphilis, chlamydiasis, they are caused by bacteria, then viral diseases which includes AIDS, hepatitis, genital herpes and genital warts, then protozoans disease includes trichomoniasis, it is caused by Trichomonas vaginalis. So gonorrhea is caused by Neisseria gonorrhea, it's a bacteria. Syphilis is caused by Treponema pallidum, and chlamydiasis is caused by Chlamydia trachomatis. AIDS caused by HIV virus, human immunodeficiency virus. Hepatitis is caused by hepatitis B virus. Genital herpes, herpes simplex virus, and genital warts is caused by human papilloma virus, HPV, human papilloma virus. These are the sexually transmitted diseases. Infertility and causes. Inability to conceive or bear children even after 2 years of unprotected sexual cohabitation is said to be infertility. The reasons could be physical or congenital, immunological or even psychological. It also includes drugs. So these are the causes for infertility either in males or females. Okay. Then there are methods to assist these infertile couples to have children. The methods which are used to assist infertile couples to have children are said to be assisted reproductive technologies, abbreviated as ARTs. These includes IVF-ET, in vitro fertilization and embryo transfer, then ZIFT, zygote intrafallopian transfer, GIFT, gamete intrafallopian transfer, IUI, intrauterine insemination, then intrauterine transfer, then AI, artificial insemination, and intracytoplasmic sperm injection. So these are the some of the assistive technologies which are used to assist infertile couples to have children. So therefore called assisted reproductive technologies. So let us take up some of the questions from this chapter. Reproductive health refers to, look at the options, a state of complete physical, mental and social well-being. This is the definition of health given by WHO, World Health Organization. So therefore this is not the right option. A total well-being in all aspects of reproduction, synapses helbekar ಹೇಳ್ತಾ ಇದೆ. So it is nothing but reproductive health. A state of absence or disease or physical fitness. This is not applicable to this question. And a state of absence of sexually transmitted disease, this is also not applicable. So therefore the right option is B. In India, family planning programs were initiated in the year. It's a direct question. Family planning programs were initiated in the year 1951 at the national level in India. Which of the following sexually transmitted diseases are completely curable? Try to remember 3H. One, hepatitis
One is HIV, it's not curable, completely curable. Then second one is hepatitis B, it's not completely curable. Then genital herpes, it's not completely curable. So andre even more options allu kuda there are diseases which cannot be cured completely. So hagadre question ಏನು ಹೇಳುತ್ತೆ, which of the following sexually transmitted diseases are completely curable? Hagadre, so this is the right option: gonorrhea, syphilis, genital warts, chlamydiasis and trichomoniasis. These are the diseases which are completely curable. This is the right option. Therefore, A is the right option. Read the following statements about contraceptives and choose the incorrect option. Contraceptives are regular requirements for maintenance of reproductive health. Of course, they are not regular requirements. So therefore, this is the wrong option with respect to contraceptives. They are practiced against natural reproductive events, right? Then they have significant role in checking uncontrolled growth of human population. This is also right. Possible ill effects should not be totally ignored. This is also right. Hagadre illi yavadu applicable agandre A, contraceptives are not actually regular requirements for maintenance of reproductive health. So therefore, this is the right option. Pick out the odd one out. LNG, it's a hormonal IUD. Waltz, it's a barrier method for female. Saheli, it's a oral pill for female. But vasectomy is for male. Andre it's a sterilization procedure in male. So ಮೂರು ಕೂಡ ಯಾವ್ದಕ್ಕೆ ಬರುತ್ತೆ ಅಂದರೆ 3 belongs to female. And these are the contraceptive methods in females, but this is in case of male. So therefore, the right option is D. Look at the next question. Which of the following is not IUD, intrauterine device? ಈ ಒಂದು questions ಅಲ್ಲಿ ಯಾವುದು IUD ಅಲ್ಲ ಅಂತ. LNG-20, it's a hormonal IUD. Okay. Then copper 7, it's a copper releasing IUD. Even multilode 375 is also copper releasing IUD, but Saheli is a new oral contraceptive pill developed by CDRI Lucknow. So which of the following is not a IUD antaga? So D is the right option. So find the incorrect statement about Saheli. It's a oral contraceptive pill developed by CDRI Lucknow. This is the right option, the right statement. It's once a week oral pill. Yes, it's also right option, statement. It's a non-steroidal preparations. Yes, it has less contraceptive value. The significance of Saheli as a oral pill or contraceptive pill is that it's non-steroidal as well as it has got high contraceptive value. So therefore, this statement is wrong with respect to Saheli. So therefore, this is the right option, that is D. The term morning after pill refers to, నేను ಹೇಳ್ತಾ ಇದ್ದೆ, emergency contraceptive pills are also called morning after pills or day after pill. Look at the reason. Emergency contraceptive pills are often called morning after pill and sometimes even day after pill because they can be used after sex to prevent pregnancy. Hagadre term morning after pill refers to emergency contraceptive. Therefore, A is the right option. The most widely accepted method of contraception for females in India is, common ಆಗಿ ನಮ್ಮ ಇಂಡಿಯಾದಲ್ಲಿ poverty is one of the reason. They may not afford to go for high contraceptive methods. Therefore, intrauterine devices are the best methods for females in India. Andre the most widely accepted methods of contraception for females in India are intrauterine devices, not the natural methods, not the barrier methods, and oral pills. So therefore, C is the right option. Which of the following is a non-medicated IUD? ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ, intrauterine devices ಅಲ್ಲಿ 3 category ಬರುತ್ತೆ: non-medicated, copper releasing and hormone releasing IUDs. ಇದು copper releasing IUD is not the right option. LNG-20, it's a hormone releasing IUD. This is also not the right option. Multilode 375 is also copper releasing IUD. So these are copper releasing IUDs. Therefore, the right option is lips loop or spirals. Andre they are non-medicated IUDs. Select the correct option showing sexually transmitted diseases which are caused by viruses. ಈ ಒಂದು ಆಪ್ಷನ್ಸ್ ಅಲ್ಲಿ ಇರ್ತಕ್ಕಂಥ ಡಿಸೀಸಸ್ ಕಾಸ್ ಬೈ ವೈರಸಸ್ ಯಾವುದರಲ್ಲಿ ಅಂದರೆ ಹೆಪಟೈಟಿಸ್ ಬಿ ವೈರಸ್, ಜೆನಿಟಲ್ ಹರ್ಪ್ಸ್ ಅಂತ ಕರಿತೀವಿ, ಜೆನಿಟಲ್ ವಾರ್ಟ್ಸ್ ಅಂತ ಕರಿತೀವಿ, ಮತ್ತು ಏಡ್ಸ್. ಈ ಎಲ್ಲ ಡಿಸೀಸು ಕೂಡ ಕಾಸ್ ಬೈ ವೈರಸಸ್. ಬಟ್ ಇಲ್ಲಿ ಬೇರೆ ಯಾವ್ದಾದ್ರು ನೋಡಿ, ಗೊನೋರಿಯಾ ಇಸ್ ಕಾಸ್ ಬೈ ಬ್ಯಾಕ್ಟೀರಿಯಾ. ಹಾಗೆ ಇಲ್ಲಿ ನೋಡಿ ಸಿಫಿಲಿಸ್ ಆಗಿರ್ಬೋದು, ಅದು ಕೂಡ ಬ್ಯಾಕ್ಟೀರಿಯಾ. ಹಾಗೆ ಕ್ಲೆಮಿಡಿಯಾಸಿಸ್ ಆಗಿರ್ಬೋದು, ಅದು ಕೂಡ ಬ್ಯಾಕ್ಟೀರಿಯಾ. ಹೀಗೆ ಬೇರೆ ಬೇರೆ ಈ ಒಂದು ಆಪ್ಷನ್ಸಲ್ಲಿ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಡಿಸೀಸ್ ಇರೋದ್ರಿಂದ, ಬಟ್ ಫಸ್ಟ್ ಆಪ್ಷನ್ಸಲ್ಲಿ ಎಲ್ಲ ವೈರಲ್ ಡಿಸೀಸ್ ಇದೆ. ಸೊ ದೇರ್ಫೋರ್ ದ ರೈಟ್ ಆಪ್ಷನ್ ಈಸ್ ಎ. The following are the reasons for infertility in males except infertility reasons may not be because of ಯಾವುದೋ infertility reasons ಎರಡರಲ್ಲೂ ನೋಡ್ತೀವಿ. ಓಕೆ, ಬೋತ್ ಅಂದರೆ ಮೇಲ್ ಆ್ಯಂಡ್ ಫೀಮೇಲ್ಸ್ ಆರ್ ಈಕ್ವಲಿ ರೆಸ್ಪಾನ್ಸಿಬಲ್ ಆಸ್ ಫಾರ್ ಇನ್ಫರ್ಟಿಲಿಟಿ ಇಸ್ ಕನ್ಸರ್ನ್. ಅಲ್ಲಿ ಇನ್ಫರ್ಟಿಲಿಟಿ ಕಾಸಸ್ ಇನ್ ಮೇಲ್ಸ್ ಮತ್ತು ಫೀಮೇಲ್ಸ್ ಅಂತ ನೋಡಿದಾಗ ಒನ್ ಸಚ್ ರೀಸನ್ ಈಸ್ ಕ್ರಿಪ್ಟಾರ್ಕಿಡಿಸಮ್. ಇಲ್ಲಿ ಇಟ್ಸ್ ಎ ಕಂಡೀಷನ್ ಇನ್ ವಿಚ್ ಓಕೆ ಟೆಸ್ಟಿಸ್ ಡು ನಾಟ್ ಡಿಸೆಂಡ್ ಡೌನ್ ದ ಸ್ಕ್ರೋಟಮ್ಗೆ ಬರೋದಿಲ್ಲ. ಸೊ ಅದು ಕೂಡ ಒನ್ ಆಫ್ ದ ರೀಸನ್ ಫಾರ್ ಇನ್ಫರ್ಟಿಲಿಟಿ ಇನ್ ಮೇಲ್ಸ್. ಆಲಿಗೋಸ್ಪರ್ಮಿಯಾ, ಲೋ ಸ್ಪರ್ಮ್ ಪ್ರೊಡಕ್ಷನ್ಸ್ ಆಗಿರ್ಬೋದು, ಏಸ್ಪರ್ಮಿಯಾ, absence of sperm productions ಆಗಿರ್ಬೋದು. ಆಲ್ ದಿಸ್ 3 reasons related to infertility in males. ಬಟ್ ಲುಕ್ ಅಟ್ ದ ಅಮೆನೋರಿಯಾ. ನೀವು ಓದಿರ್ತೀರ ಲ್ಯಾಕ್ಟೇಷನಲ್ ಅಮೆನೋರಿಯಾ. ಅಮೆನೋರಿಯಾ ಅಂದರೆ ಆಬ್ಸೆನ್ಸ್ ಆಫ್ ಮೆನ್ಸ್ಟ್ರೇಷನ್. ಆದರೆ ಮೆನ್ಸ್ಟ್ರೇಷನ್ ಆಬ್ಸೆನ್ಸ್ ಇದ್ದಾಗ ಓವ್ಯುಲೇಷನ್ ಆಗಲ್ಲ. ದಿಸ್ ಒನ್ ಆಫ್ ದ ರೀಸನ್ಸ್ ಫಾರ್ ಇನ್ಫರ್ಟಿಲಿಟಿ ಇನ್ ಫೀಮೇಲ್ಸ್. ಸೊ ಫಸ್ಟ್ 3 related to male infertility. So therefore the correct option is D. Which of the following assisted reproductive technology would you suggest for a male with low sperm count or inability to inseminate the female? ಇಲ್ಲಿ ಆಪ್ಷನ್ಸ್ ನೋಡಿ. A. Artificial insemination, gift, gamete intrafallopian transfer, zygote intrafallopian transfer and multiple ovulation embryo technology. ಓಕೆ, ಅದು ಕಾಮನ್ ಆಗಿ ನಾವು ಅನಿಮಲ್ ಬ್ರೀಡಿಂಗ್ ಟೆಕ್ನಾಲಜಿ ಅಥವಾ ಆರ್ಟಿಫಿಷಿಯಲ್ ಬ್ರೀಡಿಂಗ್ ಟೆಕ್ನಿಕ್ಸಲ್ಲಿ ನಾವು ಓದ್ತೀವಿ. ಮಲ್ಟಿಪಲ್ ಓವ್ಯುಲೇಷನ್ ಆ್ಯಂಡ್ ಎಂಬ್ರಿಯೋ ಟ್ರಾನ್ಸ್ಫರ್ ಟೆಕ್ನಾಲಜಿ ಅಂತ ಹೇಳ್ಬಿಟ್ಟು ಇದಂತೂ ಆಪ್ಷನ್ಸ್ ಬರೋದೇ ಇಲ್ಲ. ಇನ್ ಝೈಗೋಟ್ ಇಂಟ್ರಾಫೆಲೋಪಿಯನ್ ಟ್ರಾನ್ಸ್ಫರ್, ಝೈಗೋಟ್ ವಿಲ್ ಬಿ ಟ್ರಾನ್ಸ್ಫರ್ ಟು ಫೆಲೋಪಿಯನ್ ಟ್ಯೂಬ್. ಇಲ್ಲಿ ಗ್ಯಾಮಿಟ್ಸ್. ಅದಕ್ಕೋಸ್ಕರ ರೈಟ್ ಆಪ್ಷನ್ ಈಸ್ ಆರ್ಟಿಫಿಷಿಯಲ್ ಇನ್ಸೆಮಿನೇಷನ್. ಆಪ್ಷನ್ಸ್ ಕೊಟ್ಟಿರೋದ್ರಲ್ಲಿ ಇದೊಂದು ಸೂಟ್ ಆಗೋದ್ರಿಂದ ಲೆಟ್ ಅಸ್ ಸೆಲೆಕ್ಟ್ ಎ ಇಸ್ ದ ರೈಟ್ ಆಪ್ಷನ್. ಝಿಫ್ಟ್ ಅಂದರೆ ಝೈಗೋಟ್ ಇಂಟ್ರಾಫೆಲೋಪಿಯನ್ ಟ್ರಾನ್ಸ್ಫರ್ ಅಂತ ಏನು ಕರಿತೀವಿ. So it involves transfer of zygote into the fallopian tube, transfer of zygote into the uterus, transfer of ovum into the fallopian tube, transfer of sperms into the cytoplasm of the egg. ಇಲ್ಲಿ ಆಪ್ಷನ್ಸ್ ನೋಡಿ. Try to eliminate the options. Transfer of sperms into the cytoplasm of the egg is nothing but ICSI, intracytoplasmic sperm injection. ಇದು ಬರೋದಿಲ್ಲ. Transfer of ovum into the fallopian tube, ಇದು ಕೂಡ ಬರೋದಿಲ್ಲ. ಓಕೆ, ಯಾಕಂದರೆ ನಾವು ನೇಮ್ ಇಟ್ ಸೆಲ್ಫ್ ಇಂಡಿಕೇಟ್ಸ್ ಝೈಗೋಟ್ ಇಂಟ್ರಾಫೆಲೋಪಿಯನ್ ಟ್ರಾನ್ಸ್ಫರ್. ಈವನ್ ದಿಸ್ ಆಲ್ಸೋ ಬರೋದಿಲ್ಲ. ಅಂದರೆ ದಿಸ್ ವಿಲ್ ಆಲ್ಸೋ ದಿಸ್ ನಾಟ್ ಅಪ್ಲಿಕೇಬಲ್. ದೇರ್ಫೋರ್ ದ ರೈಟ್ ಆಪ್ಷನ್ ಈಸ್ ಎ, ಟ್ರಾನ್ಸ್ಫರ್ ಆಫ್ ಝೈಗೋಟ್ ಇನ್ ಟು ದ ಫೆಲೋಪಿಯನ್ ಟ್ಯೂಬ್. ಪಿಕ್ ಔಟ್ ದ ಆಡ್ ಒನ್ ಔಟ್. ಇದರಲ್ಲಿ ಯಾವುದು ಈ ಒಂದು ಗ್ರೂಪ್ಗೆ ಬರೋದಿಲ್ಲ ಅಂತ. ಅಲ್ಲಿ ಫಸ್ಟ್ ಆಪ್ಷನ್ ಪೀರಿಯಡಿಕ್ ಅಬ್ಸ್ಟಿನೆನ್ಸ್ ಅಂತ ಇದೆ, ಡಯಾಫ್ರಮ್ಸ್ ಅಂತ ಇದೆ ಸೆಕೆಂಡ್ ಆಪ್ಷನ್, ಮಲ್ಟಿಲೋಡ್ 375 ಅಂತ ಇದೆ, ಲಾಸ್ಟ್ಲಿ ಟ್ಯೂಬೆಕ್ಟಮಿ ಅಂತ ಇದೆ. ಪೀರಿಯಡಿಕ್ ಅಬ್ಸ್ಟಿನೆನ್ಸ್ ಇಟ್ಸ್ ಎ ನ್ಯಾಚುರಲ್ ಕಾಂಟ್ರಸೆಪ್ಷನ್. ಡಯಾಫ್ರಮ್ಸ್ ದೀಸ್ ಆರ್ ಬ್ಯಾರಿಯರ್ ಮೆಥಡ್ಸ್. ಮಲ್ಟಿಲೋಡ್ 375 ದೀಸ್ ಆರ್ IUDs. ಈ ಮೂರು ಕೂಡ ದೆ ಕಮ್ ಅಂಡರ್ ಟೆಂಪ್ರರಿ ಮೆಥಡ್ಸ್. ಹೌದಾ? ಹಾಗಾದರೆ ಟ್ಯೂಬೆಕ್ಟಮಿ ಇಟ್ಸ್ ಎ ಸ್ಟೆರಿಲೈಸೇಷನ್ ಪ್ರೊಸೀಜರ್ ಇನ್ ಫೀಮೇಲ್ಸ್ ವೇರ್ ಫೆಲೋಪಿಯನ್ ಟ್ಯೂಬ್ ಇಸ್ ರಿಮೂವ್ಡ್. ಸೊ ಮೂರು ಕೂಡ ನ್ಯಾಚುರಲ್ ಮೆಥಡ್ಸ್ ಆಗಿರ್ತವೆ, ಟೆಂಪ್ರರಿ ಮೆಥಡ್ಸ್ ಆಗಿರ್ತವೆ. ಇದು ಮಾತ್ರ a permanent method. It may lead to complete sterility because of a poor reversibility. So therefore the right option is D. Dear students, let's have a quick glance on the nature of our pattern of NEET and CET examination. NEET, National Eligibility Come Entrance Test. The entrance test shall consist of one paper containing 180 objective type of questions in different languages, having 4 options with single correct answer. From physics, chemistry and biology. Biology includes both botany and zoology, to be answered on the specially designed machine gradable sheet using ball point provided at the center only. So distribution of questions. Duration of the examination is 3 hours. Type of questions are MCQ, multiple choice questions. Number of questions would be 180 and each question is a 4 marks value. Physics carries 45 questions for 180 marks. Chemistry carries 45 questions for 180 marks. Biology carries 90 marks, 50% of the questions in the NEET question paper. 45 from botany and 45 from zoology, total 90 questions carrying 360 marks. Physics 180 marks, chemistry 180 marks, biology 360 marks, and total marks would be 720. Scheme of valuation. 4 marks will be awarded for every correct answer. 1 mark will be deducted for every incorrect answer from the total score, and no mark will be awarded for unmarked answer. KCET, Karnataka Common Entrance Test, KCET.
It is conducted for selecting students for engineering, pharma and farm sciences or agricultural sciences. Agricultural sciences includes BSc in agriculture, sericulture, horticulture, forestry, agricultural biotechnology, agricultural marketing, then BHSC, home science, BTech, agricultural engineering, science and technology, dairy technology, Bachelor of Fisheries, Bachelor of Veterinary Science, that is BVSC, and Animal Husbandry. Question paper pattern in biology. ಇಲ್ಲಿ 4 ಕ್ವೆಶ್ಚನ್ ಪೇಪರ್ ಇರುತ್ತೆ ನಮ್ಮ CET ನಲ್ಲಿ, ಕರ್ನಾಟಕ ಅಥವಾ KCET ಅಲ್ಲಿ. ಒಂದು ಫಿಸಿಕ್ಸ್ ಇರುತ್ತೆ, ಕೆಮಿಸ್ಟ್ರಿ ಪೇಪರ್ ಇರುತ್ತೆ, ಮ್ಯಾಥ್ಸ್ ಪೇಪರ್ ಇರುತ್ತೆ, ಹಾಗೆ ಬಯಾಲಜಿ ಪೇಪರ್ ಇರುತ್ತೆ. ಈಗ ನಾನು ಹೇಳ್ತಾ ಇರೋದು ಓನ್ಲಿ ಬಯಾಲಜಿ ಪೇಪರಲ್ಲಿ. ಕ್ವೆಶ್ಚನ್ ಪೇಪರ್ ಪ್ಯಾಟರ್ನ್ ಇನ್ ಬಯಾಲಜಿ. ಡ್ಯೂರೇಷನ್ ಈಸ್ 80 minutes. ಟೈಪ್ ಆಫ್ ಕ್ವೆಶ್ಚನ್ಸ್ MCQ. ನಂಬರ್ ಆಫ್ ಕ್ವೆಶ್ಚನ್ಸ್ ಎಷ್ಟಿರ್ಬೋದು ಅಂದ್ರೆ 60. Approximately 15 questions from first PU syllabus and major 45 questions from second PU syllabus. Each question is of 1 mark and maximum marks 60. Key tips for entrance examination. Go through the nook and corner of the textbooks prescribed by the CBSE and NCERT. ಇಲ್ಲಿ ಫಸ್ಟ್ ಇಯರ್ ಮತ್ತು ಸೆಕೆಂಡ್ ಇಯರ್ ಕೂಡ ನೀವು ನೋಡ್ಕೋಬೇಕಾಗುತ್ತೆ. First PUC and second PU syllabus should be given equal attention. Practice exemplar problems. Practice previous year NEET and KCET papers. Refer good reference books. Discuss with your teachers and clarify all subject related doubts well in advance. Seek guidance from your teachers, parents and friends. Dear students, that's all for the session. I hope you have benefited out of this program, Gnanataranga. I thank PU department for having organized such a wonderful program for the benefit of students. And I wish you all the best in the forthcoming examination. So thank you and all the best.